audio recording for this meeting has begun.
Good morning and welcome. I would first like to remind everyone to please mute your line when you are not speaking. For media and press, the FDA press contact is Jeremy Kahn. His email and phone number are currently displayed. My name is Padma Galore and I will be chairing today's meeting. I will now call the June 9th, 2021 meeting of the Pharmacy Compounding Advisory Committee to order. Dr. Takia Stevenson is the designated federal officer for this meeting and will begin with introductions.
Good morning. My name is Takia Stevenson, and I am the designated federal officer for this meeting. When I call your name, please introduce yourself by stating your name and affiliation. Dr. Bogner.
Good morning. This is Robin Bogner. I'm Professor of Pharmaceutics at the University of Connecticut School of Pharmacy.
Dr. Desai.
Good morning. I'm Samal Desai. I'm a board-certified dermatologist in private and academic practice in Dallas, Texas, and clinical faculty at the University of Texas Southwestern Medical Center.
Dr. Fensky.
Good morning. I'm Tim Fensky. I'm the Chief Pharmacy Officer for Sullivan's Pharmacy in Boston, Massachusetts.
Ms. Fusco Walker. Ms. Fusco Walker, you may be on mute. Please unmute your phone and introduce yourself. Ms. Fusco Walker, you may be on mute. So, okay, we'll come back. Dr. Galore, please reintroduce yourself.
I'm Padma Galore. I am Professor of Anesthesiology and Population Health at Duke University.
Ms. Fusco Walker, if you can hear me now, please reintroduce yourself.
Yes, Sandra Fusco Walker. I'm the consumer representative for the Allergy and Asthma Network.
Thank you. Dr. Gupta.
Good morning. This is Dr. Anita Gupta. I'm the head of Comprehensive Pain Management at Scripps MD Anderson and the head of Interventional Pain Management at Scripps MD Anderson, and also faculty at Johns Hopkins School of Medicine in Baltimore, Maryland. Thank you very much.
Dr. Gura.
Hi, I'm Kathy Gura. I'm a clinical pharmacist at Boston Children's Hospital and an assistant professor of pediatrics at Harvard Medical School in Boston.
Dr. McElhenney.
Hi, I'm Linda McElhaney. I'm the team lead compounding pharmacist for Indiana University Health System in Indianapolis, Indiana.
Dr. Patel.
Hey, good morning, everyone. This is Kuldeep Patel. I'm the Senior Associate Chief Pharmacy Officer at Duke University Hospital.
Dr. Rebello.
Good morning, everyone. My name is Elizabeth Rebello, and I'm a professor of anesthesiology and preoperative medicine at the University of Texas MD Anderson Cancer Center.
Dr. Sun.
Good morning. I'm Jeannie Sun. I'm a pharmacist and counsel at the United States Pharmacopoeia.
Dr. Veda.
Good morning. Alan Veda. I'm a pharmacist and former Executive Vice President at the Institute for Safe Medication Practices.
Dr. Emmons.
Good morning. I'm Jonathan Emmons. I'm an Associate Professor of Psychiatry at Oregon Health and Science University and the Deputy Director of Mental Health and Clinical Neurosciences at VA Portland Healthcare System.
Dr. Katzman.
Hi there, I'm a neurologist at the University of New Mexico. I'm in the departments of neurosurgery and psychiatry, nursing, and the College of Population Health. I direct the UNM Pain Center, and I direct the public health initiatives at Project ECHO.
Dr. Sandbrink.
Yeah, good morning. This is Friedhelm Sandbrink. I'm a neurologist and pain physician, the Director of Pain Management at the Washington, D.C. VA Medical Center, National Program Director for Pain Management, Opiate Safety, and Prescription Drug Monitoring Programs in the Veterans Health Administration, and academic affiliation at Uniformed Services University in Bethesda and George Washington University in D.C.
Dr. Bowie.
Yes, I'm Dr. Michael Bowie. I'm the Senior VP Regulatory Affairs at Catmon Pharmaceuticals.
Dr. Richard Green, if you can hear me.
Yes, I can. Good morning. This is Richard Green. I'm a board-certified nuclear pharmacist, Director of Radiopharmacy Practice at Cardinal Health, Nuclear and Precision Health Solutions.
I will now introduce the FDA participants. Dr. Bormel.
Hi, I'm Gail Bormel, the Director of the Office of Compounding Quality and Compliance.
Gabrielle Cosell.
Hi, I'm Gabrielle Cosell. I'm the Acting Director of the Division of Compounding Policy and Outreach in the Office of Compounding Policy and Compliance.
Dr. Lawson.
Hi, this is Rosalind Lawson. I'm Acting Branch Chief in Compounding Policy and Outreach and OCQC, Office of Compounding and Quality Compliance.
Dr. Ganley.
Yeah, hi, I'm Charlie Ganley. I'm the Director of Office of Specialty Medicine in the Office of New Drugs, FDA.
Dr. Johnson.
I'm Clinical Reviewer on the Pharmacy Compounding Review Team in the Office of Special Medicine in the Office of New Drugs.
Dr. Kazem.
Hi, I'm Suhail Qasim. I'm a lead physician in the Pharmacy Compounding Review Team in the Office of Specialty Medicine in the Office of New Drugs. Thank you.
Thank you, everyone. I will hand it back to the chairperson, Dr. Galore.
Thank you, Dr. Stevenson. For topics such as those being discussed at this meeting, there are often a variety of opinions, some of which are quite strongly held. Our goal is that this meeting will be a fair and open forum for discussion of these issues and that individuals can express their views without interruption. Thus, as a gentle reminder, individuals will be allowed to speak into the record only if recognized by the chairperson. We look forward to a productive meeting. In the spirit of the Federal Advisory Committee Act and the government in the Sunshine Act, we ask that the advisory committee members take care that their conversations about the topic at hand take place in the open forum of the meeting. We are aware that members of the media are anxious to speak with the FDA about these proceedings. However, FDA will refrain from discussing the details of this meeting with the media until its conclusion. Also, the committee is reminded to please refrain from discussing the meeting topic during the breaks. Today, we will discuss 4 bulk drug substances nominated for inclusion on the list of bulk drug substances that may be used to compound drugs in accordance with Section 503A of the Federal Food, Drug, and Cosmetic Act, also known as the 503A bulks list: melatonin, methylcobalamin, choline, chloride, and oxitriptan. For each of the 4 substances, we will hear presentations from the FDA, have the opportunity to ask clarifying questions, hear the nominators' presentations with the exception of oxitriptan and melatonin, have the opportunity to ask clarifying questions, hold an open public hearing, and have committee discussion and voting. The May 7, 2021 Federal Register notice identified the uses FDA reviewed for each of the 4 bulk drug substances being discussed at this meeting. These uses reflect those for which adequate support was provided in the nomination. In addition, the nominations and FDA's evaluations for the bulk drug substances, which are included in the briefing document posted on FDA's website, identify the proposed and reviewed uses, dosage forms, and routes of administration. The nominators of these substances have been invited to make a short presentation supporting their nomination. To the extent that the nominators' presentations include information about additional uses, dosage forms, and routes of administration, I remind the committee that these additional uses, dosage forms, and routes of administration are not part of the agency's evaluation because the nominators either did not nominate those uses, dosage forms, and routes of administration, or they were not adequately supported. The committee will also discuss a revision FDA is considering to the list of drug products that have been withdrawn or removed from the market for reasons of safety or effectiveness, the withdrawn or removed list. FDA now is considering whether to amend the rule to add 1 more entry to the list, Neomycin Sulfate. All parenteral drug products containing Neomycin Sulfate, except when used for ophthalmic or otic use, or in combination with polymyxin B sulfate for irrigation of the intact bladder. Let us begin. We will now have Dr. Takia Stevenson read the conflict of interest statement for this meeting's 503A bulks list topics.
The Food and Drug Administration is convening today's meeting of the Pharmacy Compounding Advisory Committee under the authority of the Federal Advisory Committee Act of 1972, with the exception of the National Association of Boards of Pharmacy, NABP, the United States Pharmacopoeia, USP, and the industry representatives. All members and temporary voting members of the committee are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of this committee's compliance with federal ethics and conflict of interest laws covered by, but not limited to, those found at 18 U.S.C. Section 208 is being provided to participants in today's meeting and to the public. FDA has determined that members and temporary voting members of this committee are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflicts when it is determined that the agency's need for a special government employee's services outweighs his or her potential financial conflict of interest, or when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employees. Related to the discussion of today's meeting, members and temporary voting members of this committee have been screened for potential financial conflicts of interest of their own as well as those imputed to them, including those of their spouses or minor children, and for purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, grants, contracts, CRADAs, teaching, speaking, writing, patents and royalties, and primary employment. During the meeting, the committee will discuss 4 bulk drug substances nominated for inclusion on the 503A bulk list. FDA will discuss the following nominated bulk drug substances and the uses that have been reviewed. 1, choline chloride for liver diseases, including non-alcoholic fatty liver disease and hepatic steatosis, atherosclerosis, fetal alcohol spectrum disorder, and supplementation in long-term total parenteral nutrition. 2, melatonin for the treatment of sleep disorders in patients with autism spectrum disorder, specifically children and adolescents. 3, methylcobalamin for amyotrophic lateral sclerosis, also known as ALS, pain management, peripheral neuropathy, including diabetic neuropathy, inborn errors of metabolism, also known as genetic metabolic disorders, including methylene tetrahydrofolate reductase deficiency, also known as MTHFR, hyperhomocystinemia, including conjunctive therapy in hemodialysis patients, vitamin B12 deficiency, and autism spectrum disorder. And 4, oxitriptan, also known as 5-HTP, for the treatment for patients with tetrahydrobiopterin BH4 deficiency. The nominators of these substances or another interested party will be invited to make a short presentation supporting the nomination. This is a particular matters meeting during which specific matters related to the 4 bulk drug substances will be discussed. Based on the agenda for this meeting and all financial interests reported by the committee members and temporary voting members, conflict of interest waivers have been issued in accordance with 18 U.S.C. Section 208B3 to Drs. Kathleen Gura and Sutat Liang Tunsakul. Dr. Gura's waiver involves stock holdings of an affected entity. The aggregate value of her stock is between $50,000 and $100,000. Dr. Liang Tunsakul is only sitting the choline chloride topic, and his waiver involves investment holdings in a healthcare sector mutual fund valued between $250,000 and $350,000. The waivers allow these individuals to participate fully in today's deliberations. FDA's reasons for issuing the waivers are described in the waiver documents, which are posted on FDA's website at https://www.fda.gov/advisory-committees/human-drug-advisory-committees. Copies of the waivers may also be obtained by submitting a written request to the agency's Freedom of Information Division at 5630 Fishers Lane, room 1035, Rockville, Maryland 20857, or requests may be sent via fax to 301-827-9267. To ensure transparency, we encourage all standing committee members and temporary voting members to disclose any public statements they have made concerning the bulk drug substances at issue. We would like to note that Dr. Timothy Finsky is a representative member from the National Association of Boards of Pharmacy, NABP, and Dr. Jeannie Sun is a representative member from the United States Pharmacopoeia, USP. Section 102 of the Drug Quality and Security Act amended the Federal Food, Drug, and Cosmetic Act with respect to the Advisory Committee on Compounding to include representatives from the NABP and the USP. Their role is to provide the committee with the points of view of the NABP and USP. Unlike the other members of the committee, representative members are not appointed to the committee to provide their own individual judgment on the particular matters at issue. Instead, they serve as the voice of the NABP and USP entities with a financial or other stake in the particular matters before the advisory committee. With respect to FDA's invited industry representatives, we would like to disclose that Dr. Michael Bowie and Mr. Richard Green are participating in this meeting as non-voting industry representatives, acting on behalf of regulated industry. Their role at this meeting is to represent industry in general and not any particular company. Dr. Bowie is employed by Cadman Pharmaceuticals, and Mr. Green is employed by Cardinal Health Nuclear and Precision Health Solutions. We would like to remind members and temporary voting members that if the discussions involve any other bulk drug substances or firms not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all participants to advise the committee of any financial relationships that they may have with the topic at issue. Thank you, and I will hand it back to the chairperson, Dr. Galore.
Thank you. We will now proceed with the FDA introductory remarks from Dr. Frances Gail Vormel.
Thank you, Dr. Guler. I am Gail Vormel, the director of the newly formed Office of Compounding Quality and Compliance, the FDA office primarily responsible for, among other initiatives, developing and implementing policies and compliance strategies to help assure the safety and quality of compounded drugs. Our office aims to protect patients from the risks of contaminated or otherwise harmful compounded drugs, while also preserving access to compounded drugs for patients who have a medical need for them. I would like to welcome you to the 10th meeting of the Pharmacy Compounding Advisory Committee. Today, we will discuss 4 bulk drug substances nominated for inclusion on the list of bulk drug substances that can be used in compounding human drug products under Section 503A of the Federal Food, Drug, and Cosmetic Act, or FD&C Act. This list is known as the 503A bulk list. And as mentioned before, the substances that will be discussed are melatonin, choline chloride, methylcobalamin, and oxitriptan. Some of these substances may be available as dietary supplements. As a reminder, the discussion today focuses on FDA's evaluation of these substances as bulk drug substances for use in human drug compounding under Section 503A of the FD&C Act, and is not intended to inform FDA's regulation of these substances in dietary supplements. We also note that the availability of a substance as a dietary supplement is not a criterion considered when evaluating a substance for inclusion on the 503A bulk list. Dietary supplements are regulated under a different part of the FD&C Act, and different considerations apply to the regulation of dietary supplements and drugs, including drug products compounded using bulk drug substances under Section 503A. For example, dietary supplements are intended for oral ingestion, while drugs may be intended for administration by numerous other routes of administration, such as topically or by parenteral or intrathecal injection. These different routes of administration raise very different considerations from a regulatory perspective, including safety considerations related to risk of contamination and considerations regarding systemic absorption. The reviews conducted by the agency for bulk drug substances nominated for the 503A bulk list follow the criteria described in FDA's regulations implementing Section 503A, which are separate and distinct from FDA's statutory and regulatory provisions governing the treatment of dietary supplements. During this meeting, we will also discuss whether to add an entry for certain drug products containing neomycin sulfate to the list of drug products that have been withdrawn or removed from the market because such drug products or components of such drug products have been found to be unsafe or not effective. This list, known as the Withdrawn or Removed list, implements conditions under both Sections 503A and 503B of the FD&C Act. As in previous committee meetings, we have scheduled time for the nominators to speak and time for an open public hearing after FDA's presentation on each of the 4 bulk drug substances. There will also be an open public hearing after the FDA presentation for neomycin sulfate. I would also like to use this opportunity to provide you with an update on certain developments since the committee last met in September 2018. Some of these actions affect compounders under Section 503A of the FD&C Act, such as state licensed pharmacies, federal facilities, and licensed physicians. Other actions affect those compounders known as outsourcing facilities that are regulated under Section 503B of the Act. Finally, some of the actions affect compounders under both Sections 503A and 503B. Since our last PCAC meeting in 2018, the agency has worked to establish and develop a 503A bulk list and the withdrawn or removed list under Sections 503A and 503B. In December 2019, FDA published a final rule that added 2 entries to the withdrawn or removed list. In February 2019, FDA published a final rule establishing criteria for and placing 6 bulk drug substances on the 503A bulk list. The February 2019 final rule also identified 4 bulk drug substances that FDA had considered and did not include on the 503A bulk list. In September 2019, FDA published a proposed rule identifying 5 bulk drug substances that FDA has considered and proposed to include on the 503A bulk list. FDA's proposed rule also identified 26 other bulk drug substances that FDA has considered and proposed not to include on the 503A bulk list. Another document that affects compounding under Section 503A was issued in October 2020. FDA issued a final standard memorandum of understanding addressing certain distributions of compounded human drug products for states to consider and sign pursuant to Section 503A B3B. The final standard MOU describes the responsibilities of a state board of pharmacy or other appropriate state agency that chooses to sign the MOU in investigating and responding to complaints related to drug products compounded in such state and distributed outside such state, and in addressing the interstate distribution of inordinate amounts of compounded human drug products. The agency has also been working on policy documents that affect compounders under Section 503B of the Act, including those that advance the creation of the list of bulk drug substances for which there is a clinical need for use in compounding under Section 503B, which is known as the 503B bulk list. In March 2019, the agency determined that 2 bulk substances would not be placed on the 503B list. In August 2019, the agency issued a Federal Register notice that proposed not including 9 bulk drug substances on the 503B bulk list. In July 2020, FDA published a Federal Register notice proposing to place 4 bulk drug substances on the 503B bulk list and proposing not to include 19 bulk drug substances on that list. In March 2021, FDA published a Federal Register notice proposing to add 1 bulk drug substance to the 503B bulk list and proposing not to add 4 bulk drug substances to that list. In January 2020, FDA published a revised draft guidance concerning current good manufacturing practice requirements for compounders registered under Section 503B of the FD&C Act, which are known as outsourcing facilities. In November 2020, FDA issued a final guidance on unsanitary conditions at compounding facilities that compound drugs under Sections 503A and 503B. This guidance describes examples of unsanitary conditions FDA has observed and specifically addresses drugs, including biological products, produced by pharmacies, federal facilities, and outsourcing facilities that compound or repackage drugs or mix, dilute, or repackage biological products. Notably, during the COVID-19 public health emergency, FDA published 5 temporary guidances beginning in March and April 2020 for immediate implementation to provide guidance to states and compounders under Sections 503A and 503B on issues associated with compounding drug products during the COVID-19 public health emergency. These included guidances concerning compounding of certain drugs for hospitalized patients, compounding of certain alcohol-based hand sanitizer products, repackaging or combining propofol, and use of non-standard practices regarding the use of personal protective equipment for sterile pharmacy compounding by pharmacy compounders not registered as outsourcing facilities. All of the FDA's compounding policy documents, including those just discussed, appear on the FDA's compounding website under the section titled Regulatory Policy Information. Again, thank you for your participation on the Pharmacy Compounding Advisory Committee. We look forward to a productive meeting and to continuing to work together.
Thank you, Dr. Burmel. We will now proceed with FDA presentations, starting with melatonin from Dr. Suhail Kasim.
Good morning. I am Suhail Kasim from the Office of New Drugs. I will discuss the nomination for melatonin. This slide shows the review staff involved in the evaluation of melatonin. Melatonin is a hormone nominated for inclusion on the list of bulk drug substances for use in compounding under Section 503A of the Federal Food, Drug, and Cosmetic Act, also called the 503A bulks list. It was proposed for the treatment of sleep disorders in patients with autism spectrum disorder, specifically children and adolescents in doses ranging from 0.2 milligrams up to 5 milligrams for oral administration, although the dosage forms to be compounded were not specified. Please note that the discussions of the use of melatonin in the other pediatric populations and in certain adult populations were included in the appendix section of the briefing document, which included the evaluation of melatonin as background information. However, the other uses were not considered for the overall assessment and recommendation. In conducting the 503 evaluation, FDA reviewed publicly available information to assess melatonin based on 4 criteria. Information assessed for each of the 4 criteria will be discussed. The first criteria applied was the information considered regarding the physical and chemical characterization of melatonin. Melatonin is well characterized physically and chemically and is likely to be stable under ordinary storage conditions in the solid dosage form. However, it has limited stability in water. Aqueous-based liquid melatonin products should have corresponding stability data to support their claimed expiry. Before discussing the other 3 criteria, the next few slides will go through the general pharmacology of melatonin. Melatonin is a neurohormone that plays a key role in regulating the sleep-wake circadian rhythm, and its synthesis is associated with circadian rhythms controlled primarily by the light-dark cycle. During daylight hours, the levels of melatonin are low. During nighttime conditions, plasma melatonin levels rise rapidly. Circulating melatonin levels can impact sleep patterns. The figure illustrates the circadian profile and the timing of endogenous melatonin synthesis influenced by the light-dark cycle. Melatonin production peaks in the middle of the night between 2 a.m. and 4 a.m. in the morning, from about 10 to 20 picograms per ml during daylight hours to 100 to 200 picograms per ml during the hours after sundown. Melatonin secretion is age-dependent, as shown in the right side of the figure. The peak nocturnal melatonin levels in most 70-year-olds are only a quarter or less of what they are in young adults. Therefore, melatonin secretion varies by age and depends on the time of day for peak secretions. Whereas children with autism spectrum disorders and other neurodevelopmental disorders do not have normal physiological production of melatonin. Therefore, the pharmacological effects of exogenous melatonin on the pharmacodynamic aspects of sleep in populations with neurodevelopmental disorders may not be the same as populations experiencing other primary pediatric sleep disorders. The next 2 slides discuss the clinical pharmacology of exogenous melatonin. Most of the melatonin in the circulation is inactivated in the liver after it undergoes first-pass metabolism. It is primarily metabolized by CYP1A2 and CYP2C19. So our inhibitors of CYP1A2 enzyme may increase melatonin concentrations. Exogenous melatonin has low bioavailability. approximately 15%, and it is variable as a consequence of hepatic first-pass extraction. Because the different dosage forms have different effects on absorption, distribution, metabolism, and excretion, melatonin metabolism affects the PK based on the dosage form. Next, we will discuss the safety. With regard to non-clinical safety, no major toxicities were observed in short-term acute toxicity studies. Several repeat dose toxicities have been conducted with melatonin, which did not show major toxicities. The longest study reported consists of a 26-week chronic toxicity study in rats, which showed some minor findings. These non-clinical toxicities were at much higher doses than proposed for clinical use. In a 70-day juvenile toxicity study in rats, no toxicities were seen at much higher doses than proposed for clinical use. The standard panel of genotoxicity assays did not show genetic toxicity activity, and melatonin did not have genotoxic adverse effects. In terms of the reproductive and developmental toxicity testing, some malformations were noticed in the rabbit when dams were dosed with much higher doses of melatonin than proposed for clinical use. Carcinogenicity testing has been conducted for melatonin in the rat, and increased incidence of pituitary adenomas and thyroid tumors were seen among animals treated with the highest dose when compared to control vehicles. Overall, the non-clinical testing program for melatonin was extensive and has been shown to have high safety margins at the proposed clinical doses. The Office of Surveillance and Epidemiology conducted a search of the FDA's adverse event reporting system for reports of adverse events for melatonin, in addition to a review of the medical literature for potential safety signals reported with melatonin. The FAERS reported adverse events were consistent with the known safety profile of melatonin, including information that is discussed in the literature. The reports included cases in children with autism spectrum disorder, and we will elaborate on these adverse event profiles in the subsequent slides. The FAERS reported adverse events also primarily consisted of acute reactions to short-term exposure rather than exposure data for longer-term effects. For the few serious FAERS events identified, the information was confounded by use of melatonin with other concomitantly used medications, comorbid disease states, possible drug-drug interactions, or it lacked enough details precluding the ability to determine a drug-event association. 3 patients reported using melatonin for sleep disorder in autism spectrum disorder or neurodevelopmental disorder. Melatonin was used to manage sleep. That was a reaction to the drug used to manage a comorbidity in children with autism spectrum disorder. These 3 FAERS cases highlight the medical complexities, including polypharmacy in children and adolescents with autism spectrum disorder or neurodevelopmental disorders. The figure shows single substance melatonin exposure cases documented by the United States Poison Control Centers per year from 2008 to 2020. Now, there is a trend for rising number of reports to the American Association of Poison Control Centers National Poison Data System. The observed trend may be secondary to the increased use of melatonin. The most commonly reported reasons for all calls to U.S. poison control centers were for general unintentional exposure, which accounted for 82%, and therapeutic error, which accounted for 9%. Overall, poison center calls related to general unintentional exposure were predominantly comprised of younger children, with 91% being reported in children under the age of 5 years. CARES data included 238 spontaneously reported cases that included at least one adverse event in association with the use of melatonin. Some cases listed as many as 73 different ingredients in the suspect product containing melatonin. In most of the cases, CARES cases, there was insufficient information reported, and it was not possible to determine whether a causal connection existed between the use of melatonin and the adverse events reported because of the considerable number of other ingredients in the suspect melatonin product, or that melatonin was used with other medications, concomitant medications. The table shows pooled adverse events in controlled clinical trials associated with melatonin for primary or secondary sleep disorders in children and in adults. The daily melatonin doses reviewed in these systematically reviewed studies ranged from 0.15 milligrams up to 12 milligrams. Adverse events were characterized as generally infrequent, mild or moderate severity, and were either self-limiting or resolved on withdrawal of treatment. These included daytime sleepiness, dizziness, hypothermia, decreased appetite, and restlessness. The first column shows the number of studies these events were noted in, and the second and third columns show the events that were recorded for these particular adverse events. This table shows safety data from the double-blind randomized placebo-controlled study in patients with autism spectrum disorder who had not shown improvement after standard sleep behavioral intervention. For the pooled melatonin doses, 2 milligrams, 5 milligrams, or 10 milligrams, during the double-blind period, the AE rates compared to placebo were for somnolence, there was 28% of event rate reported versus 12%. For fatigue, 25% versus 18%. And for agitation, 18% versus 10%. So these were some of the events reported in the double-blind control studies, shown in the first two columns over here. During the 91-week long-term safety follow-up on study, shown in the columns on the right side of the table, similar type of adverse events that were reported during the double-blind study were also observed during that open-label study period. The treatment-related somnolence and fatigue were considered to reflect the pharmacological effect of residual daytime melatonin, most probably secondary to a higher dose. It was also possible that some of these participants had a differential response and were poor metabolizers of CYP1A2 enzyme, developing daytime somnolence or fatigue owing to melatonin accumulation. Because endogenous melatonin levels in humans sharply decline just before the onset of puberty, there may be a potential risk of delayed sexual maturity and with gonadal development in prepubertal children taking melatonin over extended periods. And animal studies have supported this theory. Although, in the long-term study that was discussed in the previous slide, in a subset of those children and adolescents with autism spectrum disorder, there was no reported delay in sexual maturation or pubertal development with orally administered prolonged release or controlled release melatonin after 2 years of continuous use. Melatonin appears to be relatively safe and well tolerated for oral administration at the nominator's proposed dose 0.2 milligrams up to 5 milligrams in children and adolescents with autism spectrum disorders. Before discussing the effectiveness data for melatonin, that is the third criteria in the 503 evaluation, I'll spend a few minutes on the next two slides. Disorders of sleep are one of the co-occurring medical conditions in patients with autism spectrum disorder, and it can also worsen the core symptoms of autism spectrum disorders. Sleep disorders are common and severe in children with neurodevelopmental disorders like autism spectrum disorders, compared with typically developing children. Sleep disorders in children and adolescents with autism spectrum disorders include difficulties initiating and maintaining sleep, frequent and prolonged night awakenings, irregular sleep-wake patterns, short sleep duration, and early morning awakening. For the evaluation of effectiveness of melatonin on sleep disorders in pediatric patients with autism spectrum disorders, the following sleep outcomes were considered: sleep onset latency, night awakenings, and total sleep time. In the middle section of the forest plot figure shown here, there is a meta-analysis of the controlled trials in the systematic reviews, show that the treatment effect estimates were all in the direction showing benefit with melatonin. in the patients with autism spectrum disorders who experience sleep disorders for the sleep diary reported sleep onset latency outcome regardless of the dosage form of melatonin when used in the short term. Melatonin decreased sleep onset latency with median duration 28 minutes and a pooled mean difference reduction of by 35 minutes. Similarly, in the upper section of the forest plot figure shown here, melatonin increased sleep duration or total sleep time with median duration 33 minutes and pooled mean difference 64 minutes. Please note that in this figure, the studies favor the outcome for melatonin is on the right side. The improvements in total sleep time while on melatonin treatment appeared to be due to earlier sleep onset. Although not shown here, there was no improvement in the number of awakenings per night. Adding behavioral intervention to melatonin treatment resulted in a better treatment response on the outcomes. In conclusion, melatonin may be effective for the short-term treatment of sleep disorders in children and adolescents with autism spectrum disorder under the supervision and care of healthcare practitioners. The treatment response in favor of melatonin for the pharmacodynamic sleep outcomes evaluated may vary based on the melatonin dose, the timing of administration, and the dosage form. Lastly, we apply the fourth criterion, that is the historical use of melatonin in compounded drug products. Melatonin has been used in pharmacy compounding since at least 2009 and has been compounded in a variety of dosage forms. According to the Johns Hopkins University CERSI report, melatonin is sometimes used as a sleep aid in patients with autism spectrum disorder in the United States. Melatonin is approved for use in Europe, Australia, and Japan. We have balanced the four criteria that was discussed to evaluate melatonin for the 503A bulk list. Based on the information that we have considered, a balancing of the four evaluation criteria weighs in favor of melatonin for oral administration being added to the 503A bulk list. Thank you.
Thank you, Dr. Kassim. We will now take clarifying questions for FDA presenters. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and the end of your follow-up question with "that is all for my questions," so we can move on to the next panel member. With this, I would like to invite the questions. I see Dr. Emmons has raised his hand. If you could speak.
Hi, this is Jonathan Emmons. Thank you, Dr. Kassim. I think my only question is, and I don't know if you are prepared to comment on this, but could you talk about the magnitude of the effect in terms of increases in total sleep time and decreases in sleep latency relative to kind of other treatments for insomnia?
Dr. Gullo, can I respond to the question?
Yes, please.
Dr. Emmons, thank you for that question. First of all, I have to acknowledge that we do not, as you may be aware, there are no FDA-approved products for pediatric use. There are products that are being used off-label, that is FDA-approved products that are being used off-label for use in the pediatric population. But in terms of comparative efficacy for when used along with melatonin, I was not able to come across any data to indicate, except from the recommendations for the Academy of Neurology and the Academy of Pediatrics, which made recommendations including discussions for melatonin, but I did not come across any information that was specific in relation to other products that are being used for managing sleep disorders. I hope that answers your question. Thank you.
Thank you, Dr. Kasim. Dr. Emmons, I see your hand is not raised anymore, and we'll assume that you have no further questions.
Thank you. I have no further questions.
I do not see any other raised hands. So with that, we will conclude this section on clarifying questions for the presenters. I would like to state into the record that we do not have a nominator presentation for this topic. I would also like to state into the record that there are no open public hearing speakers for this topic. With that, the open public hearing portion of this meeting has now concluded, and we will no longer take comments from the audience. We will now give another opportunity for remaining clarifying questions for all the melatonin presenters. Please use the raise hand icon to indicate that you have a question, and remember to put your hand down after you have asked your question. Please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. As a gentle reminder, it would be helpful to acknowledge the end of your question with a thank you and end of your follow-up question with "that is all for my questions," so we can move on to the next panel member. At this time, I do not see any raised hands for further clarifying questions. With that, we will move into the panel discussion questions part of the meeting. Would the panel members like to discuss the topic and present their views? Seeing as there is no discussion, the committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments, which were not present for this particular in the open public hearing. We will proceed with the question to the committee and panel discussions for melatonin. I would like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. Today's question is a voting question. Dr. Takia Stevenson will provide the instructions for the voting.
Question 1 is a voting question. Voting members will use the Adobe Connect platform to submit their votes for this meeting. After the chairperson has read the voting question into the record and all questions and discussion regarding the wording of the vote question are complete, the chairperson will announce that voting will begin. If you are a voting member, you will be moved to a breakout room. A new display will appear where you can submit your vote. There will be no discussion in the breakout room. You should select the radio button, that is the round circular button, in the window that corresponds to your vote, yes, no, or abstain. You should not leave the no vote choice selected. Please note that you do not need to submit or send your vote. Again, you need only to select the radio button that corresponds to your vote. You will have the opportunity to change your vote until the vote is announced as closed. Once all voting members have selected their vote, I will announce that the vote is closed. Next, the vote results will be displayed on the screen. I will read the vote results from the screen into the record. Next, the chairperson will go down the roster, and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. Are there any questions about the voting process before we begin?
As there are no questions, I will now read the question in hand. FDA is proposing that melatonin for oral administration be included on the 503A bulks list. Should melatonin for oral administration be placed on the list? Do voting panel members, have any questions about the wording of the question itself? Seeing none. If you vote no, you are recommending FDA not place the bulk drug substance on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes subject of an applicable USP or NF monograph or a component of an FDA-approved drug. If there are no questions or comments concerning the wording of the question, we will now begin the voting on question for melatonin. Dr. Veda, I see you have a question.
Yes, my only question is that this is just for oral administration, correct?
Correct. That is what the question states, only oral administration. Does that answer your question, Dr. Veda?
Yes, it does. Thank you.
Thank you. Do we have any other questions? Since there are no questions or comments concerning the wording of the question, we will now begin the voting on question for melatonin.
We will now move voting members to the voting breakout room to vote only. There will be no discussion in the voting breakout room.
Sub-conference rooms are open.
Voting has closed and is now complete. Once the vote results display, I will read the vote result into the record. Voting has closed and is now complete. The vote results are displayed. I will read the vote totals into the record. The chairperson will go down the list and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. There are 13 yeses, 0 noes, 0 abstentions.
Thank you. We will now go down the list and have everyone who voted state their name and vote into the record. You may also provide justification for your vote if you wish to. We'll start with Dr. Gura.
Kathleen Gura, I vote yes.
Dr. McElhaney.
Oh, Linda McElhaney, I vote yes.
Thank you. Dr. Desai.
Hi, Samal Desai. I voted yes, and I appreciate the FDA's clear presentation on the data.
Thank you, Dr. Desai. Padma Gulor, I voted yes. Dr. Rebello.
Elizabeth Rebello, I voted yes.
Dr. Fensky.
Tim Fensky, voted yes.
Dr. Bogner.
Robin Bogner, I voted yes.
Dr. Sun.
Jeannie Sun, I voted yes.
Dr. Emmons.
Jonathan Emmons, I voted yes.
Dr. Veda.
Alan Veda, I voted yes.
Dr. Patel.
Khalid Patel, I voted yes based on the data presented by the FDA.
Thank you. Dr. Gupta.
Anita Gupta, I voted yes.
Fusco Walker.
Sandra Fusco Walker, I voted yes.
Thank you, everyone. We will now take a 15-minute break. We will reconvene at 11:55 Eastern Time for the methylcobalamin topic. Panel members, please remember that there should be no chatting or discussion of the meeting topics with other panel members during the break. Thank you. Welcome back, everyone. Welcome back, everyone. Welcome back. We will now proceed with the FDA presentation on methylcobalamin from Dr. Susan Johnson.
Hello, can you hear me?
Yes, we can hear you now.
Thank you. Well, good morning, My name is Susan Johnson, and I'm a clinical reviewer in the Pharmacy Compounding Review Team. I'll be discussing the FDA review of methylcobalamin. There we go. I'd like to thank the methylcobalamin primary review team and give special thanks to the clinical review staff in several therapeutic review divisions in the Office of New Drugs. Representatives of each division listed on this slide have joined us to help should questions arise about their medical specialty. Methylcobalamin has been nominated for inclusion on the list of bulk drug substances for use in compounding under Section 503A of the FD&C Act. It was reviewed for use in the treatment of vitamin B12 deficiency, various inborn errors of metabolism, hyperhomocystinemia, autism spectrum disorder, amyotrophic lateral sclerosis, and various peripheral neuropathies and pain conditions. The proposed dosage forms include oral, sublingual, nasal spray, and injectable solution for subcutaneous or infusion administration. Turning now to the physical and chemical characterization of methylcobalamin. Methylcobalamin is one of 4 structurally related vitamers of vitamin B12. Each vitamer has a different ligand attached to the same cobalamin base. This is a table for reference comparing selected features of the various cobalamins. Methylcobalamin is a naturally occurring substance in humans and animal proteins. Methylcobalamin is not a component of FDA-approved products. The B12 vitamers hydroxycobalamin and cyanocobalamin both are in FDA-approved products, primarily for use via intramuscular injection, although there is an approved nasal spray product. Each of the vitamers is available in oral dietary supplements, and as we heard earlier, today's discussion does not address dietary supplement products. Methylcobalamin is stable at room temperature in solid formulations when protected from light. As an aqueous solution, it is stable when protected from light and stored at a controlled temperature with a controlled pH. We conclude that methylcobalamin is well characterized and, if formulated and stored appropriately, would be stable in solid and liquid dosage forms. Moving now to safety, beginning with general pharmacology. When orally ingested, all of the cobalamins are initially separated from proteins to which they may be naturally bound, and then attached to the protein haptocorin. As they move to the duodenum and ileum, they are dissociated from haptocorin and become attached to a second protein commonly referred to as intrinsic factor. The complex is then moved into ileal endocytes via a receptor-mediated calcium-dependent endocytosis. Once absorbed into the systemic circulation, all cobalamins are transported through the plasma in an inactive state. attached to transcobalamin or haptocorrin. Passive absorption of cobalamins also occurs throughout the gastrointestinal tract. The model on this slide was compiled by its authors from a literature review of the pharmacokinetics of cobalamins. It shows that each type of cobalamin, including methylcobalamin, reaches the cell wall in an inactive state. Cobalamins are then absorbed into the cytosol, where the unique ligands are removed and each is reduced to the base cobalamin molecule. The overall process to reduce each to the base molecule is the same for each cobalamin, although the kinetics and enzymes involved are specific to each. From the base cobalamin molecule, active methylcobalamin and adenosylcobalamin are formed. The multiple metabolic steps in this diagram are controlled by enzymes and cofactors whose actions may be altered due to inborn errors of metabolism that we will discuss later. In the center of this diagram, B12 represents active methylcobalamin. Active methylcobalamin is a cofactor for methionine synthase in the reaction that methylates homocysteine, converting it to methionine. Methionine is in turn important in generating S-adenosyl L-methionine, or SAM, and then SAMe, that are critical to DNA and RNA synthesis. Non-clinical pharmacokinetics suggest a saturable absorption process and renal excretion, which are consistent with human pharmacology. Acute toxicity studies show it safe to be given at high doses for short periods of time. But other elements of a full non-clinical safety program were not found in the literature. We found limited clinical pharmacokinetic data for methylcobalamin. Oral bioavailability is inversely proportional to the consumed dose. A small amount of cobalamin from dietary intake, amounting to several milligrams, is stored in the liver, and only 0.1 to 0.2% of that is lost per day due to metabolic activity. Turning to clinical safety information, the clinical studies of methylcobalamin that we reviewed provided limited adverse event information, but several studies included reports of non-serious adverse events. We looked at the labeling of the FDA-approved cobalamin products, as they may provide safety information that is relevant to methylcobalamin based on the apparent comparability of their pharmacologic action. Although the most common reactions listed in labeling for hydroxycobalamin and cyanocobalamin products are non-serious, the approved drugs have been associated with serious and fatal events, and we find that these may occur with methylcobalamin as well. FAERS data showed 174 reports of serious adverse events. 2 cases reported use of methylcobalamin alone, including one of apparent jaundice in a 3-year-old receiving methylcobalamin. Neither case reported sufficient information to establish causality. CARES data regarding use of dietary supplement products included 384 reports. Among these, there were 6 deaths reported. No reports appear to have sufficient information to assess causality, and most are confounded by use of multiple ingredient products or multiple concomitant products. There were 2 cases in which methylcobalamin was the only product reported to have been used. In conclusion regarding safety, methylcobalamin is found in foods, and its use orally as a dietary supplement does not appear to be associated with serious adverse events. Serious adverse events that are listed in FDA-approved labeling for injectable hydroxycobalamin and cyanocobalamin products could occur with methylcobalamin based on the comparability of mechanism of action. Overall, we have a concern regarding lack of available safety data with methylcobalamin, particularly for intravenous injections and infusions. Turning now to the effectiveness factor in the review, the first use we looked at was vitamin B12 deficiency. This condition used to be commonly associated with causing pernicious anemia, but methyl B12 deficiency is now seen mainly in older patients with age-related changes in the function of gastric parietal cells or intrinsic factor, or in individuals with low B12 intake levels, such as vegans. One observational study of the use of oral methylcobalamin in children was found from India, where methylcobalamin is more commonly used than the cobalamins approved in the U.S. This study reported the correction of low cobalamin levels in 27 of the 28 children dosed orally for 1 month with methylcobalamin. In conclusion regarding the effectiveness of methylcobalamin for the treatment of vitamin B12 deficiency, the pharmacologic mechanism of methylcobalamin appears comparable to that of FDA-approved cobalamin products, and we find that it's likely to be effective in treating vitamin B12 deficiency. We also found limited clinical data to support its use in oral form to treat vitamin B12 deficiency. However, no data were found that show hydroxycobalamin or cyanocobalamin injectable products are insufficient to provide treatment for vitamin B12 deficiency when an injectable product is required. We next reviewed the use of methylcobalamin to treat inborn errors of metabolism. These are caused by genetic mutations. There are numerous inborn errors of metabolism that affect absorption, transport in the blood, intracellular metabolism, or physiologic actions of the cobalamins. Some of these disorders, such as methylmalonic aciduria, are responsive to chronic supplemental administration of hydroxycobalamin or cyanocobalamin. No studies of the use of methylcobalamin in these disorders were found. Methylene tetrahydrofolate reductase, MTHFR deficiency, and MTHF dehydrogenase deficiency are inborn errors of the folate cycle. They are treated with folic acid and betaine. Classic homocystinuria is an inborn error of metabolism that decreases the conversion of a homocysteine to cysteine. It's treated with hydroxycobalamin. No clinical studies were found that support the use of methylcobalamin in the treatment of these inborn errors of metabolism. The next use we reviewed is hyperhomocystinemia. This is not an inborn error of metabolism and can result from a variety of causes, including the use of certain drugs such as metformin. 85% of patients with chronic kidney disease have hyperhomocystinemia. Current practice guidelines for these patients do not recommend routine use of folate or vitamin B12 except in cases of deficiency. We found no specific data to support the effectiveness of methylcobalamin to treat hyperhomocystinemia. Looking at the use of methylcobalamin to treat autism spectrum disorder, or ASD, we found 4 published studies. 2 studies were uncontrolled, and each included approximately 40 patients. Due to lack of a control arm, neither provides substantive information about whether methylcobalamin is effective in ASD. In the first of 2 controlled studies, no statistical differences were demonstrated between the methylcobalamin and placebo groups. In the second controlled study, there was a statistically significant difference between methylcobalamin and placebo on a nonspecific measure of clinical improvement. But this finding was not associated with differences between the treatment groups with respect to behavioral scales specific to ASD symptoms. These studies do not support the effectiveness of methylcobalamin to treat ASD. The Johns Hopkins CIRCE study will be discussed again later with respect to use of methylcobalamin in ASD patients. To assess effectiveness, key opinion leaders were queried and responded that B12 would have no effect in ASD unless being used to treat a dietary deficiency. We conclude that we found no specific data to support the effectiveness of methylcobalamin in treating ASD. The next use that we reviewed was amyotrophic lateral sclerosis, or ALS. Based on the results of a post-hoc analysis of a phase 2-3 study conducted by a Japanese pharmaceutical firm, the same firm decided to undertake another study. We have not found that the results of the second study have been published. We conclude that we were unable to identify studies that show methylcobalamin is effective in the treatment of ALS. Studies of the use of methylcobalamin in the treatment of various types of neuropathy were analyzed for within rather than between group responses, were subject to bias per the author, or failed to show statistically significant differences between methylcobalamin and placebo. The available data do not support methylcobalamin's effectiveness in the treatment of peripheral neuropathy. Studies of methylcobalamin to treat pain either failed to show differences between treatments or did not analyze differences between active and placebo groups. Available data do not support methylcobalamin's effectiveness in the treatment of pain. In conclusion regarding effectiveness, the pharmacologic mechanism of methylcobalamin suggests that, like FDA-approved cobalamin products, methylcobalamin is likely to be effective in treating vitamin B12 deficiency. But we found no data regarding clinical situations in which the FDA-approved cobalamin products are insufficient to provide treatment when an injectable treatment is needed. We found negligible or no specific information to support effectiveness of methylcobalamin to treat inborn errors of metabolism, hyperhomocysteine, ASD, ALS, peripheral neuropathy, or pain. Turning to the fourth factor of use in compounding, FDA requested a review from the University of Maryland, Searcy. They found little information to characterize methylcobalamin's use. Johns Hopkins Searcy was asked to look at methylcobalamin use in patients with autism spectrum disorder. Data were derived from a clinical sample of nearly 1,800 patients at the Kenny Krieger Institute, a registry population sample, and a Medicaid claim search. Estimates of the portion of ASD patients who use methylcobalamin ranged from less than 1% to 6% of ASD patients. FDA found online promotions from compounding pharmacies and treatment clinics promoting methylcobalamin for a wide variety of conditions and diseases, such as depression, Alzheimer's disease, AIDS, and ASD. A number of different routes of administration were described, including intravenous infusions. In summary, we consider methylcobalamin to be physically and chemically well characterized if formulated and stored properly. I want to highlight considerations related to the other factors. Methylcobalamin is a vitamer of vitamin B12, so based on pharmacology and the use of FDA-approved cobalamin products, methylcobalamin would be expected to be effective in treating vitamin B12 deficiency. However, it's unclear that methylcobalamin provides a unique benefit over the other vitamers of B12 that are available in FDA-approved products. It is also unclear that the treatment of vitamin B12 deficiency is currently the primary use of compounded injectable methylcobalamin in the United States. It appears that the primary use is to treat patients with conditions, in some cases serious, for which there is little evidence to support the effectiveness of methylcobalamin. In these situations, we do not have information on the range of doses used or the frequency of administration, and we find we cannot make a judgment regarding the safety of the current use of injectable products in patients. Therefore, we find that a balancing of the 4 evaluation criteria weighs against methylcobalamin being added to the list of bulk drug substances that can be used in compounding under Section 503A of the FD&C Act. Thank you. I'm happy to take questions.
Thank you, Dr. John. We will now take clarifying questions for FDA presenters. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and the end of your follow-up question with "that is all for my questions," so we can move on to the next panel member. With that, I would request that the summary slide for methylcobalamin that was just presented be displayed. The slide before, 30, slide 30.
Hello?
Thank you. If we could display slide 30, and once that is done, I would request Dr. Patel, who has a question, to speak.
Hello, this is Kuldeep Patel. Thank you, Dr. Johnson, for that presentation. I just wanted to better understand a little bit more of the two controlled studies that were summarized. I believe it was mentioned that the difference was not statistically significant, so I just wanted to know a little more in depth of what, at least one of the two controlled, it seemed like there might have been equivalency, but we didn't get a chance to kind of understand the details of what was being compared and what were the endpoints that were being looked at. Thank you.
Sure. Do we want to pull up slide 22, please? Thanks. So a couple of, oopsies.
Susan, I can operate your slides for you. This is Takia Stevenson speaking. I can move your slides for you. Just one moment. I got it. One moment. Go ahead, Susan. You can speak.
Oh, thanks very much.
Sure.
Great. Thanks very much. So let's look at the details. One of the things that I would point out about these studies is that they're relatively small, and had they found a lot of effect in ASD, it would still make it perhaps hard to judge whether they were generalizable. to the entire ASD population. But in the Bertoglio study, and I think we have folks who are connected with these studies in the nominator presentation, so we may be able to ask more questions about the actual occurrence that maybe aren't even in the publications. But I would point out that the Bertoglio study was a double-blind, placebo-controlled crossover study of 30 children, aged 3 to 8. They received 6-week treatment with 64.5 micrograms per kilogram of methylcobalamin, and they received it subcutaneously every 3 days, and then they received the alternate treatment. So there were multiple endpoints studied, including a fairly commonly administered clinical global impression improvement scale. And I can list for you the other scales. I'm not sure if that's something that you would be interested in. The second study was a double-blind, placebo-controlled study in 57 children, ages 3 to 7. They were treated for 8 weeks with 75 micrograms per kilogram or placebo, and so approximately, every 3 days. There was a within-group improvement in CGI that was statistically higher for the methylcobalamin group than placebo. But no difference on 2 other behavioral scales that assess the ASD specific aspects. And those were the parent-rated ABC or the social responsiveness scale. I'm not an expert on these scales, and we have subject matter experts in ASD who could perhaps comment further if there were specific questions about what was looked at or what was found.
I think it would be helpful, this is Khalid Patel, it would be helpful to know if that improvement scale is sort of the standard measure that is used for better understanding effectiveness of treatment options in this disorder.
Sure. So we have online Dr. Horn and Dr. Rossetti from the Division of Psychiatry. And Dr. Rossetti would like to speak to this question.
Yes, can you hear me?
Yes, we can. Thank you.
All right. So yes, I'm Dr. Rossetti from the Division of Psychiatry. So regarding the first study, Bertoglio from 2010, they used as outcome measure, beside the CGI-I, the clinical global impression improvement, they used the aberrant behavioral checklist, the ABC, that is the usual scale that we use also in the agency. Plus they use other 4 different scales, the parent interview for autism clinical version, the Peabody picture vocabulary test, and then MacArthur communication development inventory. And when they look at this scale, all the behavioral scale and the improvement scale, they were not able to identify any difference compared to placebo, as Dr. Johnson said. The second study, Hendron study from 2016, they used 3 outcome measurements. One was, again, the clinical global impression improvement scale, and the other 2 were again the aberrant behavioral checklist scale and also the social responsiveness scale. So they were not able to find any significant difference between placebo and methylcobalamin in the behavioral scale. Regarding the CGI high scale, when they look at that as a percentage of improvement, that is, so they treat it like a categorical variable, that is usually what we do, they did not find any significant difference. When they look at it as a continuous variable, they will be able to find this significant difference of a 0.7 point. It actually is less than 1 point. That is what is required to move from one category to the other one. Like from the CGI, 4 is no improvement, and 3 would be mild improvement. Did I answer your question?
Yes, ma'am. Thank you.
Thank you.
Thank you, Dr. Patel. Does that answer your questions? Do you have any further questions?
No, that was very helpful. Thank you.
Thank you. I do have a question for the specialist who was just speaking about the various scales as a follow-through. You know, again, not being an expert, but looking up the CGI and how it's conducted, it appears there's no universally accepted scoring guideline for the 7 points. Rather, it sounds like it was based solely on clinical judgment and, you know, seems to go with clinical gut sense about how the patient is doing. How is this being used in research in a standard way to compare between, you know, considering that it could be clinician bias?
All right. Thank you for the question. So this is a very important point, and we agree with you. Usually, for example, the 2 drugs that have been approved by the FDA for autism spectrum disorder, it is a combination of the ABC scale and the CGI high scale. Only the CGI high scale is not enough. Did I answer your question? Okay.
Yes, it does. Thank you very much. I appreciate it.
Okay.
Do we have, we do have Dr. Desai who has questions, if you would speak.
Thank you, Dr. Galor. I appreciate the FDA's thorough presentation and walking us through this, as well as the input from our specialist on the call. My question is more related back to the beginning of Dr. Johnson's presentation and stability. It seemed like one of the concerns based on what was presented was the IV formulations. I'm just trying to get a sense of when weighing these factors that have been presented, was the stability one of the major concerns from the FDA team when reviewing this?
This is Sue Johnson. It was not a primary concern for us, largely because we think that this is a manageable formulation issue, and that if proper steps were taken in terms of light, pH, temperature, a formulation that could work could be generated by compounding pharmacies. We can talk a little bit more.
Did we lose Dr. Johnson?
Dr. Johnson, we're not able to hear you. Dr. Johnson, are you on mute? We will give Dr. Johnson a few minutes to reconnect.
Hello?
Dr. Johnson, are you reconnected?
Yes. Hi, I've changed phones. Dr. Ganley's letting me use his setup. I don't know why it flipped me out, but it wouldn't let me dial back in with the same number. So we're
Not
back.
a problem. We're glad to have you back.
We're back. So the main point about the formulation issues is that we did not find that to be a significant consideration in recommending that methylcobalamin not be added to the 503A list. We think that the formulation issues could be overcome by compounding pharmacies.
Thank you, Dr. Johnson. And I think what I'm just trying to ascertain, because there was such a large breadth of points made in your very nice presentation, what would you say is the main concern for this not to be included?
Thanks for that question. Yes, there were a lot of nominated conditions and a lot of different types of data sets, so it is difficult to coalesce into one recommendation. But I think our primary concern is that while we do feel that methylcobalamin should act in the same pharmacologic manner as the other cobalamins and therefore be effective for treating vitamin B12 deficiency, its use, and I think we'll hear some more about this in the nominator presentations, does not seem to be related to treatment of vitamin B12 deficiency primarily. It seems to be being used in a number of other conditions and not as an oral product, but as an injectable product, as subcutaneous, IM, intravenous, and intravenous infusions. Those we just feel we don't have a lot of safety information on, practically none. One of the things that you'll find in the literature is that methylcobalamin is used much more in Asia and Australia. It just isn't used as much as the other FDA-approved cobalamin, so we have more data on those. But the main consideration is the way it's being used is something that we do not have a lot of safety information about.
Thank you, Dr. Johnson.
Such as, and I would just add, such as the range of doses, frequency of doses, exactly what treatments people are receiving.
Thank you, Dr. Johnson. Dr. Desai, did that answer your question?
Yes, thank you, Dr. Galore.
Dr. Ganley, I see that your hand is raised. Would you like to speak?
Oh, he raised his hand, so he would be recognized for me to get back in.
I see. Thank you very much. I do not see any other hands. I just see Dr. Katzman. I see your hand is raised.
Yes, thank you. Yeah, this is Joanna Katzman here. Dr. Johnson, thank you so much for your great presentation. Your last comment about the uses of methylcobalamin for indications outside of the United States, predominantly the studies done in Asia for kind of neurological conditions was very helpful. In fact, I've seen some recent studies for subacute postherpetic neuralgia and more remote studies for kind of chronic post-thoracotomy pain that actually look, fairly decent, although, like you said, they need more data and they haven't really been done here in the U.S. My question to you, Dr. Johnson, is wanting to know if you've seen any studies looking at comparing methylcobalamin with the other types of cobalamin to any kind of crossover studies with the conditions listed to look for any efficacy or safety issues that might prohibit the use of methylcobalamin for low B12 or effectiveness in any of the indicated conditions?
That's an interesting question, and part of where our review started was to find out if, in fact, methylcobalamin added to the armamentarium for B12 products, and we did not find comparative information that would allow us, and I think the point in the presentation is we don't have information that shows the FDA-approved products are insufficient. And we did not find comparative information that showed that methylcobalamin, with the exception of vitamin B12 deficiency, would be effective for the same indications that the approved cobalamins are used for. Does that answer your question? Yeah, we had hoped to find that as well, but we did not.
Right. Can I just ask one more question in a little bit of a different way, Dr. Johnson? Are there any contraindications to using methylcobalamin for nutritional repletion in patients with vitamin B12 deficiency? In addition to the other two cobalamins?
My understanding, and I'm going to let our experts speak on this. My understanding is if one treats pernicious anemia too aggressively, one can cause permanent neurologic damage. And that is the only significant treatment caution that I have seen associated with it. But I'm going to ask from the Division of Non-Malignant Hematology, Dr. Ryan or Dr. Dieseroth, if you'd like to add to any limitations to the use of B12 in treating vitamin B12 deficiency. Sorry, use of methylcobalamin in treating vitamin B12 deficiency.
Thank you.
I think Dr. Ryan is allowed to participate as a responder.
Dr. Ryan, the chair recognizes you if you'd like to speak. Dr. Ryan, are you on mute?
I think we can work behind the scenes to get an answer and come back, perhaps?
That would be fine, Dr. Johnson. We will move on to the next panel member with questions unless, Dr. Katzman, you have further questions?
No, thank you so much, Dr. Johnson.
We will we'll try to get Dr. Ryan online and have her address the question.
Wonderful. Thank you, Dr. Johnson. And Takia will let me know and we will recognize Dr. Ryan when ready. The next panelist who has their hand raised for a question is Cisco Walker. Would you like to speak?
Yes. Thank you very much. Dr. Johnson, I'm wondering if you can explain what the difference risk would be for a person who would use a compounded injectable version of the methylcobalamin instead of an FDA-approved product?
So just to be clear, methylcobalamin is not in any FDA-approved products.
Right, one of the other two, I mean, I'm sorry.
And I think I'm going to ask Gail Bormel to talk in general about FDA's viewpoint about using FDA-approved products versus compounding products, but again, to keep in mind that we wouldn't be talking about products that contain the same active ingredients. So Gail Bormel, would you like to comment?
Sure. Is that okay, Dr. Goulart?
Yes, of course. Please.
Okay. So just, yes, and as Dr. Johnson said, you know, we're just talking about, in general, FDA-approved drugs versus compounded drugs by state licensed pharmacies, federal facilities, or, you know, licensed physicians. So generally, FDA-approved drugs, you know, are subject to pre-market review, so they're looked at in terms of safety and efficacy before they are marketed. And also, FDA-approved drugs are made according to CGMP conditions, you know, the CGMP regulations. In contrast, compounded drugs are not reviewed for safety, efficacy prior to marketing, and they are not, the compounded drugs that are made in accordance with Section 503A of the Act are not subject to current good manufacturing conditions. So there's differences in review and in the quality conditions that compounded drugs under Section 503A are subject to versus FDA-approved drugs. Does that answer the question?
Yes, thank you for clarifying that. I have no more questions.
Thank you, Dr. Bormel, and thank you, Ms. Cisco Walker. I understand you have no more questions. We will move on to the to Dr. Fensky, who had his hand raised. Would you like to speak?
Hi, thank you. Tim Fensky. Dr. Johnson, thank you for the presentation. I just need to clarify one thing that I heard from, whether you can clarify it or the expert can clarify this for me. So basically, when we're talking about the ASD and the studies that were being held, I heard something about the CGI-I scale is not the only scale that needs to be assessed when doing this, and correct me if I'm wrong, and that's why the other scales are being used. And if that's the case, does the FDA identify any products that do make a significant improvement to these scales for the core symptoms of ASD?
I can answer this. I'm Dr. Rasetti, the Division of Psychiatry. So yes, we have two drugs that have been approved so far for autism spectrum disorder, and one is the risperidone, and the other one is the aripiprazole. They are not approved for core symptoms of ASD. They have been approved for irritability. The scales that have been used are two. One is the CGI, the CGI. The second one is the ABC, the Aberrant Behavior Checklist. That is a 56-item scale that has different sub-items, including irritability, social withdrawal, stereotypic behavior, hyperactive non-compliance, inappropriate speech. And for the risperidone and for the aripiprazole, this scale showed a significant improvement regarding the subscale of irritability. And for the risperidone, they did two, I think there were 3 double-blind placebo-controlled studies, and for the aripiprazole, there were 2 double-blind placebo-controlled studies, and both of them show significant difference in this scale. Did I answer your question?
Yes, thank you very much. I have no further questions.
Thank you.
Thank you both. Dr. Fensky, you have no further questions, and I see that your hand has been lowered. With this, at this time, I do not see any further panel member questions, clarifying questions. So we will move on. We will now proceed with the nomination.
I think we might have Dr. Ryan on the phone. Dr. Ryan, are you available now? Dr. Ryan?
Dr. Johnson, we will have an opportunity after the nominator presentations for further questions and during the discussion. Perhaps we can have her join us then.
Certainly. Thanks very much.
Thank you. We will now proceed with the nominator presentations. We have two presentations. Dr. A.J. Day, who is speaking on behalf of National Community Pharmacists Association and Alliance for Pharmacy Compounding, followed by Dr. Richard E. Fry, who is speaking on behalf of Professional Compounding Centers of America. Dr. Day.
Good morning or good afternoon, depending on where you're listening in from. I do not have the red bar that gives me control over the slides. Okay. Thank you very much. Well, good day, and thank you for inviting me to speak to this committee today. My name is A.J. Day. I am the Vice President of Clinical Services at PCCA. I serve on the board of the Alliance for Pharmacy Compounding and with the NTPA Compounding Committee. And I'd like to express my gratitude to begin with to the FDA for hosting this virtual meeting, which in this format allows for greater participation from stakeholders, particularly physicians, than we have had in the past. Our nomination is specifically focused on patients with autism spectrum disorders. We will not be presenting on other nominated uses. I'd like to start by addressing this excerpt from FDA's analysis, stating that it is not clear that methylcobalamin provides a unique benefit over other vitamins of B12 that are FDA approved. This statement should actually be flipped around. There is no evidence that other vitamins of B12 are safe or effective in patients with autism spectrum disorders. All clinical literature for the use of B12 in patients with ASD focus on the use of methylcobalamin. To remove this option and suggest that patients and providers should use cyanocobalamin or hydroxycobalamin, which, as Dr. Johnson pointed out, are not naturally occurring in humans, while methylcobalamin is, will force clinicians to experiment on these patients. There is no data to guide dosing, no data on safety, no data on clinical outcomes using these other vitamins, not even anecdotal data. FDA discusses the process for activating exogenously administered cobalamins. This process relies on fully functioning gastrointestinal lining, transport proteins, and intrinsic factor. Dr. Johnson did an excellent job of walking us through all of that process. A hallmark feature for many patients with ASD is compromised gut and intestinal health and sensitivity to proteins and foods. Therefore, oral absorption of cobalamins may not be the same in ASD patients. Data assessed by Paul and Brady 2017, as referenced by FDA, as well as their reference citations, does not explore altered gastrointestinal health or function in patients with ASD. Now, Paul and Brady actually acknowledge that very point in their paper shown in this excerpt on your screen, where patients with impaired production of intrinsic factor, such as autoimmune pernicious anemia or atrophic gastritis, and or a compromised intestinal absorptive function, as in celiac disease, ulcerative colitis, Crohn's disease, or tropical sprue, may greatly impair B12 absorption by endocytosis. The research provided by Johns Hopkins University acknowledges their perspective of only using methylcobalamin to address vitamin deficiencies and therefore predominantly utilizing oral dosing. Given the altered GI status of this patient population, it is unsurprising that patients generally do not show significant improvement in that environment. Presentations by Dr. Fry and Dr. Newbrander later today will address the biochemical and clinical reasons for subcutaneous dosing of methylcobalamin is preferred for patients with ASD. It is also important to acknowledge that detailed reviews by FDA and Johns Hopkins did not identify significant safety concerns. There are many trials published for various potential uses, including ASD, and FDA states that adverse events reported with methylcobalamin use are infrequent and non-serious. Methylcobalamin is an approved product as an injection in both Japan and Australia. Safety data from the Japanese product indicates an adverse reaction rate of 0.45%. The product warns of potential anaphylactoid reactions, though no such reactions have been documented. Similar data is on the Australian product label. Additionally, Australia's Therapeutic Goods Administration has a searchable database of adverse event notifications for medicines. Searching for all single-ingredient methylcobalamin reactions from January 1, 1991 through March 1, 2021, there are no results for adverse reactions related to the injectable product. There are 4 non-serious case reports related to oral formulations with no details provided for other health conditions or medications that patients may have had. Therefore, causality cannot be determined. Several publications are available which report adverse events. This first portion of the table lists articles which assessed subcutaneous methylcobalamin in patients with ASD. This next slide includes other indications that were studied. This data is included as extra data on the safety profile of injected methylcobalamin, even when used IV at relatively high doses at a frequency that exceeds the commonly utilized dosing schedule for ASD. Articles highlighted in yellow were not included in FDA's bibliography. Dosing in blue highlights milligram as opposed to microgram dosing protocols. Here we have additional studies, and when we look at the totality of the data from all of these trials in these 3 slides, with their combined 531 weeks of trial data, the only serious adverse events were identified in the trial for ALS, and those were not attributed to the methylcobalamin therapy. When we consider therapies for patients with ASD, we must remember that these patients receive high-frequency, in-depth medical supervision with a multimodal care approach. Current clinical trial designs are generally ill-suited to measure changes in core symptoms of ASD. This article from the National Institute of Mental Health states that the gold standard diagnostic instruments were not created to measure severity or improvement of the disorder, and they do not holistically and comprehensively measure changes in core symptom domains. FDA does point out that there are no approved drugs to treat the core symptoms of ASD, and the only 2 drugs approved to treat symptoms of irritability associated with autism have significant adverse events, including EPS. An informal survey of compounders 2 weeks ago reports that 24 pharmacies responded as dispensing compounded methylcobalamin to a combined 27,565 patients in the last 12 months. 662,000 milliliters of compounded methylcobalamin was dispensed by those pharmacies to those patients in that 12-month time period, keeping in mind that the typical dose is 0.1 mLs. This request in the survey was specific for data representing patients with ASD. Medicaid data on utilization of methylcobalamin provided by Johns Hopkins research is not informative. Prescription claims for compounded methylcobalamin are unlikely to be submitted to CMS because CMS generally does not reimburse for it. Surveys by University of Maryland and JHU key opinion leaders are severely limited, and no physicians that we have spoken with were contacted by either research organization. We have no information about how physicians were selected to participate or why the numbers of physicians selected were so few. In May of 2017, FDA hosted a patient-focused drug development meeting for autism. This URL listed on the slide takes you directly to the transcript of that meeting, where FDA officials, physicians, family and caretakers of patients with autism and autism spectrum disorders discussed the shortfalls of clinical trial designs for this patient population, the impact of methylcobalamin injections, and various FDA-approved products and therapies. Here we have a screenshot of a comment that is submitted to the public docket from a physician specializing in chronic illness in children and adults with ASD. I hope you all have the opportunity to read this and the multitudes, hundreds of other comments that were submitted. This is simply highlighting a specific comment within the previous slide that discusses the biochemical pathways that injected methylcobalamin addresses in the patients that he treats. I'm limited on time for this presentation, so I will not be reading the specific comments and the details of that. Similarly, Dr. Buckley points out specific clinical outcomes achieved with injected methylcobalamin. Additionally, the Medical Academy of Pediatric Special Needs identifies compounded injectable methylcobalamin as a standard of care. In conclusion, injectable methylcobalamin is commercially available in Japan and Australia. There are no significant adverse events identified from global literature searches, and FDA states that adverse events reported with methylcobalamin use are infrequent and non-serious. It has been compounded since at least 2005. There are no alternative options for patients with ASD. Denying methylcobalamin from the 5038 bulks list will eliminate therapy for thousands of patients where no significant safety risk has been identified. There are no products, FDA approved or otherwise, that address the core symptoms of ASD to even the same degree as compounded methylcobalamin reported in the presentation from Dr. Johnson. As Ms. Bormel stated in her introductory remarks, the goal of this process is to protect the public from unsafe compounded drugs while maintaining patient access to needed compounded therapies. No evidence has been presented to establish compounded methylcobalamin as unsafe, and there are significant numbers of patients who need continued access to this therapy. Dr. Fry will address the clinical and biochemical assessment. Thank you very much.
Thank you, Dr. Day. Dr. Fry, if you'd like to start your presentation.
Thank you very much for inviting me to talk here. I'm presenting on behalf of PCCA. I'm Chief of Neurodevelopmental Disorders and Director of the Autism Program at Phoenix Children's Hospital. I have been treating children with autism almost exclusively for the past 15 years, mostly from a biochemical point of view. Another medical point of view, and I'd just like to say that methylcobalamin injections I found is one of the most effective treatments for some children with autism, with some having very significant response and significant positive response to the treatment. So today I'll talk a little bit about methylcobalamin treatments for autism spectrum disorder. Just my disclosures, several research grants, industry funding, and advisory boards to disclose, and my disclaimer, of course, this is not an FDA-approved treatment when we talk of it as treatment. I have put some key points here, not enough time to go through them all, but in general, what I'd like to talk about is the biochemical pathways that are abnormal in children with autism and how methylcobalamin appears to be very therapeutic in helping fix these pathways, and how when we talk about the double-blind placebo-controlled studies that there was some discussion about, we see that there is evidence in those studies that the children that did have response biochemically did improve their behavior and symptoms. So what we find in children with autism is very interesting. We find abnormalities in these four interconnected pathways. That is, problems with methylation, redox metabolism, folate metabolism, and tetrahydrobiopterin metabolism. We find these are all connected, and there's many groups that have studied this, and what I've highlighted in red in this diagram are the metabolites and the enzymes that have been shown to be abnormal in multiple studies from multiple groups, so it's been verified. What I also have here are these green boxes which show us where we can intervene into these biochemical pathways, and we can see that B12 is very key for intervening into methionine synthase. One of the first people to really identify these biochemical abnormalities was Jill James at Arkansas Research Institute, and she was able to find in multiple studies and confirm this, that children with autism had abnormalities in both methylation and redox metabolism, and at that point she suggested it was a subgroup of children with autism. And studies have looked at this data, and there is a nice meta-analysis and systematic review in Free Radical Biology and Medicine for those that are interested, that shows that these abnormalities are repeatable and shown by many groups. What's also been shown is that there's abnormalities in glutathione, the major antioxidant of the body, as a result of these abnormalities in multiple tissues in the body, including the brain. Also, what's been shown is that these redox abnormalities result in oxidative damage to DNA, proteins, and lipids in children with ASD. These abnormalities seem to be pretty pervasive, and recently Dr. Jill James teamed up with Jürgen Hans at the Roswitha Polytechnic Institute, where they used discriminant analysis to ask whether this is a signature of autism, these abnormalities in methylation and oxidative stress. And indeed, they found in their first study that looking at these metabolites of abnormal methylation and oxidative stress, that you can actually discriminate children with autism for those that are typically developing with about a 98% accuracy. So Dr. James was really the first one to show that the simple treatment of injected methylcobalamin 3 times a week and oral folinic acid daily improved these biochemical markers of methylation metabolism and oxidative stress, including improvement in the production of glutathione. In that study, it was an open label study. It was shown that the glutathione redox ratio, which is shown in B, improved in many, but not all of the children. In this study, the Vineland Adaptive Behavior Scale was performed at the beginning and at the end of this 3-month study. We took that data to ask if there was any improvement associated with this treatment, and we found that the change in months in development ranged anywhere from 2 months to 12 months on average, and many skills actually improved anywhere from 6 to 12 months in 3 months of the treatment with methylcobalamin. And these were kids that were not making progress, so wouldn't even be expected to change much on these scales. Of course, this was an open label study, so one of the things that we wanted to see is whether the biochemical changes correlated with the behavioral changes. And we did find that for 6 of the 9 subscales that there was a significant association with improvement in biochemical pathways, with improvement in behavior and symptoms. So this is the double-blind placebo-controlled study done by Dr. Robert Hendren, one of the 2 double-blind placebo-controlled studies of methyl B12. This study did not use folinic acid or folate with the methyl B12, which I think could be a significant difference from the previous study. They randomized 57 children. And as mentioned before, there was a significant improvement overall in the clinical global impression scale improvement at the 0.005 level. The other scales did not show improvement in those that were treated. However, it should be noted that both the social responsiveness scale and the ABC, the Aberrant Behavior Checklist, as just previously noted in this paper by Palmer, are parent-reported scales. And what we found in clinical trials with individuals with autism is that parent-reported scales are very much open to large placebo effects. But one of the things that I think is very important about the Hendren study, even though not all of the outcomes were positive, is that the change in the CGI, the Clinical Global Impression Scale, was correlated with improvements in the methylation capacity, that is the SAM-SAR ratio. So again, improvements in the biochemical pathways were correlated with improvements in the Clinical Global Impression Scale. This was also true of the previous double-blind placebo-controlled study, where overall there was no significant effects, but in a subgroup that did improve, there were biochemical changes. One of the things that we know about children with autism is that the disorder is very heterogeneous, and that there may be certain subgroups that respond to certain treatments and have certain etiologies. So it's very important to understand when you're looking at autism spectrum disorder that there are many subgroups that we have to find that will respond to different treatments most likely. And I believe that the studies that we've shown here suggest that there may be this subgroup, which may be significant, that responds to methyl B12. Overall, the clinical studies, again, there's 2 double-blind placebo-controlled studies, there's 2 prospective open-label studies, and there's several case reports. All of them show improvement in ASD symptoms. Again, the double-blind placebo-controlled studies and one of the open-label studies show that these improvements are correlated with changes in biochemistry, and there was few adverse effects, and when they occurred, they were very mild. How does B12 work? Well, we talked about improving function of these biochemical pathways, particularly to improve methylation and glutathione production. But one of the other ways that we know that improving this pathway helps the brain is that glutathione is made of glutamate. And one of the things that is a theme in autism is there's too much hyperexcitability and too much glutamate in the brain of children with autism. And by reducing glutamate, you can rebalance the excitatory-inhibitory ratio. Other evidence from this study, for example, by Jill James, demonstrated that there are certain polymorphisms that are associated with autism within these pathways, particularly the transcobalamin B12 binding protein. And if an individual has a polymorphism in the B12 binding protein and in COMT, they are at significant risk for having autism with an odds ratio of 7. There has also been case reports or case series of 4 patients with a novel TCN2 mutation. This is the B12 binding protein, and 2 of them had autism. Interestingly, in these cases of children with autism, when they were treated with chloride, they actually developed acute anemia, which was only improved when they were given methylcobalamin. And methylcobalamin was shown to improve their symptoms. Also, there's evidence of decreased levels of B12 in the brain of children with autism. Methylcobalamin in particular was 3.1 times lower in the brain of individuals with autism as compared to controls. We also find that the biochemical abnormalities that would be associated with a decrease in B12 and has been also found in individuals with autism were also found in the brain, including changes in methylation and glutathione metabolism. In addition, we find that there's decreased expression of the mRNA from methionine synthase in the cerebral cortex of those with autism, and then it decreases much faster with age than typically developing populations. Also, there is evidence that dietarily there is lower B12 intake in children with autism spectrum disorder, as shown in this meta-analysis. And this is another letter that you did see, which I'll highlight, from the Medical Academy of Special Needs president, that states that this is a treatment that is considered standard of care for children with autism through their training program, and they find that it's extremely effective, and this is an organization of over 1,000 physicians that treat children with autism. And I would say that one of the problems with getting funding, I am an academic researcher and trying to get funding for researching B12, and one of the comments on 2 of the grants that were not funded were that methylcobalamin was standard of care and widely used. So it wasn't seen to be novel to actually fund for further clinical research because it was already something that was already widely used. And with that, I will thank you for giving me the time to present this to you.
Thank you, Dr. Fry. We will now take clarifying questions for nominated presenters. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and the end of your follow-up question with "that is all for my questions," so we can move on to the next panel member. I would actually like to start the questions, and I do see we have other interested panel members as well. This question is for you, Dr. Fry, if you could answer. You have stated that the downsides, shall we say, of vitamin B12 injections are minimal, and that there are no real known side effects. I'm curious as to your view on the 2010 paper by Geyer, an autism cohort study, where they found cobalt levels following vitamin B12 injections were significantly high. And just to stress, the dose was 75 micrograms per kilogram, and what they summarize over here is that methylcobalamin injections significantly increased the mean levels of plasma cobalt 6.83-fold and urinary cobalt 51-fold in comparison to unexposed subjects, and significant positive correlations were found between the frequency of methylcobalamin injections and the levels of plasma and urinary cobalt. And our concern, of course, being that cobalt toxicity affects the heart and a few other things, but the cardiomyopathy. I'd also, as a follow-up, like to clarify on the Dr. Day presented that one patient with an MI where it was considered not related to the methylcobalamin. So we'll start with Dr. Fry. If you could comment on that study and your thoughts on the cobalt toxicity with injectables.
So again, I have patients that have been on methylcobalamin injections for long periods of time, and I have not seen any toxic effects that would be linked to cobalt. Although I understand your concern. The fact that there's elevated levels, and sometimes we see even elevated levels of B12 in the blood. And we actually found this was linked to something called the folate receptor autoantibody in one of our studies. So one of the things that we take as a kind of an overarching notion is that the children with autism have metabolic systems that are under stress, so they need higher levels of these vitamins for those systems to work properly. So the fact that they're elevated in the blood just in general is not concerning, because we're treating a disorder, and the levels may not, for normal individuals, may not be relevant as much to individuals with autism. That is, for example, one of the other areas that we've made a lot of progress in is that we find that folate has trouble getting into the brain, and we know that the mechanisms behind this, one of the things that we can do is increase folate levels in the blood to actually push the folate through the reduced folate carrier into the brain and restore levels. And this has been measured.
Dr. Fry, I apologize for interrupting, but my question was very specific to cobalt toxicity and increased cobalt levels with 75 micrograms per kilogram of vitamin B12 being injected. I'm summarizing that your response was that you have anecdotally not seen this in the study, in the patients that you've treated per se, but that you would also be concerned that cobalt toxicity could be a possibility?
I think that it's something of further study, but I don't see any evidence that it seems to be harmful to my patients. In fact, I see many positive effects, and when we stop the B12 injections, as sometimes we do, to see if they're necessary, after a certain amount of treatment, you find that children start to lose skills, they feel tired, they regress a bit. So it seems like the treatment seems to give them more benefit than side effects. And I have not seen any side effects of cobalt. Although, being an academician, I would think that anything's important to study, because, that's something that, we have a question about. So, I think the question is very insightful and valid, but I don't see, and I haven't heard from any of my colleagues, this concern or any symptoms that would make me think that there's cobalt toxicity.
Thank you, Dr. Fry. So just to summarize my read on this study, which included 72 patients, so it was not larger than some of the other studies presented so far on the benefits. The other question was, there was a paper that I would appreciate your comment on, which was on speech disorders in autism patients, and they reviewed multiple substances that could help in that arena. And it's a summary report that was done. I apologize, I'm going to pull the reference up for you in just a second. But essentially, that paper on speech disorders, which reviewed multiple substances, close to 12, that could help patients with autism spectrum disorder, summarized that the oral formulations were safe and effective, which seems contrary to what we've just heard, where oral formulations of drugs are less useful in autism spectrum disorder. So I would appreciate your view on the fact that there's a published review with multiple other experts who seem to feel like oral formulations are safe, and that anything else would require larger clinical studies to ensure safe and efficacious dosage, which is similar to the cobalt side effect study, which also states that larger studies are required to establish the safe dosage of intravenous methylcobalamin.
Okay. Well, first, this isn't intravenous. This is subcutaneous.
Subcutaneous.
So, yeah. So if the study, the cobalt study, was intravenous, that would definitely be a difference. But the studies that have used methylcobalamin orally have all been combined with other supplements. They do show that there is some benefit overall of of multivitamins that include cobalamin. However, I would caution that many children with autism don't tolerate oral. First of all, anything oral. They are very restricted, so we can't give them anything oral. And two, many children have severe GI problems, including inflammation in the gut, which may limit the ability to absorb B12 and to produce intrinsic factor, probably. So they may not be absorbing it if we give it, orally. And just in general, what's associated with autism is severe food restriction. So we need these other compounds injected in other formulations so that these substances can be given to children with autism and they can receive it. And I have that issue with many patients.
Thank you. I do appreciate your difficulty as an academician in obtaining research funding for this. That said, it appears that at least a few papers in this arena all call for larger translational medicine studies to be performed so safe new protocols to treat these ASD-mediated disabilities can be introduced. Would you agree that there is a need for controlled studies and a larger scale?
I do, most definitely, and that's what we're currently doing with leucovorin right now because it seems to be so helpful for some children. We have gained funding for doing multiple multi-center clinical trials, and I completely agree that that would be great. The problem is, a practical matter, this area of research in autism is very underfunded, and I don't know that people realize how underfunded and how much impact we could make if we were able to research these substances. And just briefly, one of the comments in the first grant I submitted was your exact questions about the oral form of the and whether it was superior, that is injected versus superior. And so in the second grant, we actually included a separate arm that included the oral form of methyl B12. And I think the reviewers were a little bit confused on the non-inferiority analysis, and it was a little bit too complex for them at that point. That with the idea that, they thought this was kind of standard of care, I think was prevented us from getting funding.
Dr. Fry, thank you very much for your detailed answers. Much appreciated. With that, I would like to request Dr. Wada to speak on his questions.
Yes, thank you very much. And thank you, Dr. Day and Dr. Fry, for those presentations. I just have a quick question. It seems the FDA did have an issue with lack of reports on safety. And unfortunately, that's because the 503As don't have to report adverse events, yet it seems like the drug is drugs being compounded. First, for Dr. Day, you said there was 27,000 patients that were treated with 0.1 ml. The other question that the FDA had was the range of doses. So for both of you, what was the common dose with those 23,000 or 27,000 patients? Or do you know?
This is AJ Day. The most common concentration of the compound is going to be 25 milligrams per ml with a dose of 0.1 ml per dose. That is most common. There are going to be some interpatient variabilities, and Dr. Fry and as well as our speaker in the open public hearing, I believe, will address some of those as well. Dr. Fry?
Yes, I think that that you're absolutely correct. It is very variable, and that's a real area where we need to study more. The clinical trials used anywhere from about 60 to 75 micrograms per kilogram. But I find in my clinical practice that it varies considerably, and many times you have to titrate the dosing anywhere from about will be 1,500 to probably 3,000 micrograms every other day to every 3 days as far as injectables.
All right, thank you. No further questions.
Thank you. Dr. Gora?
Thank you. This question is directed to Dr. Fry. First, excellent overview of the situation. My question is, with the fact that there are products available from Japan and Australia, are you aware of any attempts of anybody trying to import them to do studies rather than using a compounded product? And is there any sort of registry for autistic patients to record their responses to therapy so we can get that data that was asked about common doses and adverse events? Thank you.
Yeah, that's such a great question. No, I'm not aware of anybody trying to get the formulations from outside the states. And the question about the registry is very very good. There's been several attempts and several, what we would say, surveys of the use of certain treatments in autism to see which ones seem to be the most effective with the least side effects. And they have shown, and they've all pointed to methyl B12 as being one of the most effective treatments with the lowest amount of side effects. Right now, we're working on a national survey that Dr. Jim Adams did with, I believe, about somewhere between 5,000 respondents. And we found that for children that were on methyl B12 injectables, they had a significant improvement over time in the parent-rated severity of their disorder compared to children that didn't take that treatment. And it was perceived, it's one of the treatments that were perceived as most effective by parents. Of course, this is all most of these surveys are retrospective, which is, unfortunate because, there's limitations in that type of research. And unfortunately, there's no prospective registry. At this point, I know that there's several groups, including Autism Speaks, who is trying to develop a network where we standardize the way we see patients, so at least clinically we can record their improvements over time, as well as any types of changes that they may have received from therapies, both positives and negatives.
Thank you. No further questions.
Thank you. We will now begin the open public hearing session. Both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the advisory committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement, to advise the committee of any financial relationship that you may have with the product, and if known, its direct competitors. For example, this financial information may include the payment by a bulk drug supplier or compounding pharmacy of your travel lodging or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. The FDA and this committee place great importance in the open public hearing process. The insights and comments provided can help the agency and this committee in their consideration of the issues before them. That said, in many instances and for many topics, there will be a variety of opinions. One of our goals today is for this open public hearing to be conducted in a fair and open way, where every participant is I apologize, I seem to have been muted. Technical difficulties. I will repeat the last sentence. That said, in many instances and for many topics, there will be a variety of opinions. One of our goals today is for this open public hearing to be conducted in a fair and open way, where every participant is listened to carefully and treated with dignity, courtesy, and respect. Therefore, please speak only when recognized by the chair. Thank you for your cooperation. Speaker number 1, your audio is connected now. Will speaker number 1 begin and introduce yourself? Please state your name and organization you are representing for the record.
This is Dr. Newbrander. Can you hear me?
We can hear you, Dr. Newbrander. I apologize. There seems to be some technical echoes.
Yeah, it's feeding back. I don't think it's my phone, so I don't know.
Dr. Newbrander, this is Takia Stevenson speaking. Can you hear me? Dr. Newbrander, can you hear me?
Can you hear me now?
Yes, we can hear you now.
I don't know why it's feeding back. Is there something that you knew how to do on your side?
May I ask you, maybe you might have to mute your computer speakers and speak just into the phone only. That might get rid of the echo.
Computer speakers. Can you hear me now?
Yes, that is much better. Thank you.
Great. Let me, I'm Dr. Newbrander from New Jersey. I have no financial interest, or nobody pays me for anything, which is sad. So anyhow, that's a joke. Nobody pays me for my trips or me to prescribe this or anything else, so I have no financial interest. Let me say this, though. I was trained as a pathologist, and that study on the cobalt toxicity, it has nothing to do with my 7 minutes I'm going to talk, but it came up. The problem is when you test for cobalt, you know, in cobalamin, the cobalt is in the center of cobalamin, and it's tied in there. It's not biologically active. And when you have to test to see what cobalamin is, you have to burn it off, and it goes through, and it comes off. So when you put it in, when you take the blood or the urine, whatever you take, and you put it into the instrument, you have to burn it off. And now you expose that it's there, but biologically it's inactive. So that's the answer to that. All right, let me just say here, let me just do this. I'm going to just, okay. All right, will you advance the slides for me, because I have no way to do that. Okay, let's go to the first slide. Basically, keep on this slide. The first slide said that the FDA has what we've been hearing about, questions about safety, effectiveness, adverse effects, and dosing. On this second slide, it's extremely important. This is the 2016 brain tissue study by Zhang and Deep, and it showed that methyl B12 is present in much higher amounts during the early stages of development and the reproductive years. Here you can see that children with autism had 1/3 the amount of methyl B12 than did those from infancy to 20 years of age. So this amount that the children with autism have is essentially that of a 50-year-old. Next slide. Dr. Richard Deese, a well-known researcher in the field of methylation, he shared the importance of continuing injectable methyl B12 for children with autism. First, he discussed that methylation is impaired in autism and associated with oxidative stress, and monitoring these identifies 98% accuracy of those with autism. Dr. Fry mentioned that also. Next slide, please. Next, Dr. Deep reiterated that methyl B12 is only 1/3 that of normal levels, that normalizing brain levels can improve neurodevelopment in ASD, and he stated that the other forms of B12, like cyanocobalamin, are not efficiently converted to methylcobalamin. Next slide, please. He ended his discussion by saying the blood-brain barrier restricts entry of cobalamin into the brain, indicating high dosage levels are needed to augment brain levels of methylcobalamin. He went on to say clinical trials have demonstrated injectable methyl B12 produces benefits very quickly, the example shown being a significant drop in the ATEC score after only 8 weeks. He concluded by stating that high doses of methyl B12 may be adding clinical benefits beyond its cofactor role. Next slide, please. Currently, only risperidone and aripiprazole are approved medications for autism, specifically to decrease irritability. By contrast, methyl B12 improves many autistic symptoms. On May 26 and again June 7, I provided the FDA with a list of 83 of the commonly observed symptom improvements we see, 197 pages of parental quotations in my practice, 541 pages from the evaluation tool I use in my clinic that documents undeniable symptom improvements from injectable methyl B12. And I have estimated that over the years I have received, after taking away placebo effect, 33%. After taking away that, I have estimate I have over 6 million words from parents documenting what methyl B12 has done for them and what's different in my clinic. It's in the first 6 to 9 weeks with no changes. Next slide, please. Since May 2002, when I discovered the effect of methyl B12 for autism, I have personally prescribed and monitored for at least 3,500 children, more than 1.5 million doses, with excellent results in a few weeks with no serious adverse effects. A very conservative estimate. I just gave an estimate here, but Dr. Day gave better criteria. I said at least 25 million doses have been done without significant effects, and his is higher than that, which that's good. Next slide, please. This early study from my clinic documented that the ATEC scores improved significantly within 6 to 9 weeks when the children did not make any other changes to anything they were doing for any treatment other than methyl B12 injections. Next slide, please. A later study from my clinic with an N of 321 and a p-value of less than 0.0001 again showed excellent clinical improvements in just 8 weeks when the children did not make any other changes to anything they were doing whatsoever other than methyl B12 injections. Next slide, please. Here you will see the form I use. In addition to ATEC, the parent-designed report form shown here is an additional evaluation tool I use in my clinic. It does not replace ATEC, but for some symptoms, many symptoms really, it is more sensitive to changes in a child. One of the major differences in what I do in my clinic and the evaluation tools is parents have to document in detail with examples what the child was like before they started every 3-day shots and what the child is like after they started the shots. And again, they have to give detailed examples and whatever. And so by giving before and after with detailed examples, it takes away the placebo effect. Basically here, from this list of 83 symptoms of methyl B12, those above are the ones that more than 40% of the parents have observed in 6 to 9 weeks after making no changes to anything they were doing other than methyl B12 injections. Side effects are misunderstood. Many of the side effects from methyl B12 are because the processing speeds in the autistic brain are considerably slower. By studying the graph in the 2009 publication by Thatcher that studied 54 autistic children from my clinic The processing speeds in autistic children's brains were slowed 150% in the central region, 185% in the frontal region, and 275% in the occipital parietal region of the brain. The precuneus and cuneus in the parietal region are involved in organization, sensory integration, synthesis, and manipulation of auditory, visual, and kinesthetic input, and it's slowed 275% in autism. Deeper portions in the parietal lobe are involved in memory and quick decisions in crisis situations, again slowed 275% in kids with autism. The occipital cortex is involved with memory encoding, with semantic task and visual association, and again it's slowed 275% in autism. So therefore, each desirable methyl B12 symptom improvement has generated new brain waves that must be processed near the parietal center of the parietal area, and it's slowed down 275%, so parents see sensory issues there. However, the remaining overflow, unprocessed excess stimuli have to go to the motor cortex, and it's slowed down 150%, so the parents see hyperactivity and stimming. The additional unprocessed excess stimuli overflow to the frontal cortex. It's slowed down 185%, so parents see irritability and worse behaviors. These types of side effects are not bad, but part of the process. No different than the pain of an operation. I didn't tell you to advance your slide. I'm so sorry. Please advance the slide. Oh, sorry. Another one. I'm so sorry. Those are the 80-some symptoms. That's where that was where I said it slowed down. Please advance it again to the next one. That one's the precuneus. That's where it is. This is where it has that. I'm so sorry. One more slide.
That's okay. Just a reminder that it's been 8 minutes of your time.
I know. Okay, let me just go through this real quickly. Okay, basically, this is the side effects. Okay, let me advance another one real quickly. So I can, okay, that's what I was talking about. Advance again to the side effects. Okay, these are the net side effects. You can look at them. I've given you all this information in detail with my script. Okay, the next side effect, the slide, please. The types of side effects, there are several, and they're very uncommon, so go ahead. Next slide. I'm almost finished. This one's important, though. Basically, here you'll see. This is the dependency, etc. The serum levels for B12 in autism is not the pattern of a B12 deficiency, rather a B12 dependency. Zhang's paper documented 1/3 less methyl B12 in the brains of autistic children. Deeth alluded to autistic children having high-dose serum B12 dependency when he said high-dose treatment with methylcobalamin may reflect additional beneficial actions beyond its cofactor effect. I only got a couple more real fast slides, real fast. Next one. Basically, subcutaneous, we've done all of them. People come to me from all over, literally 80 countries, and I've seen over 3,500 kids, and when they come to me on oral, a lot of them come on oral methyl B12. It hasn't done anything to any significant degree. We put them on the subcutaneous and does great. 2 more slides real fast for real. Next one. The calculated dose, you can read about that. And here, basically, I believe that as a group, Dr. Day, Dr. Fry, and myself have established that there is safety, that there is effectiveness, that there is a lack of medication for these kids, and that methyl B12 can fill that void. So therefore, we all hope, and all my patients, we all hope that we've established that it would be unconscionable to take methyl B12 injections away from children on the spectrum and their families. Thank you.
Thank you. Speaker number 2, your audio is connected now. Will speaker number 2 begin and introduce yourself? Please state your name and any organization you are representing for the record.
Hello, my name is Heather Volk. I'm an associate professor in the Department of Mental Health from the Johns Hopkins University Bloomberg School of Public Health. I have nothing to disclose regarding relationships with methylcobalamin or B12 or related substances. The data that I am going to verbally present to you today, and I apologize for my scratchy voice, I'm a bit under the weather and a late addition to the agenda, so I have not sent slides, is all available publicly on the Johns Hopkins University Center for Excellence in Regulatory Innovation our Johns Hopkins CERCI Center website. The report is posted there. Briefly, over the past 2 years, we conducted a study to examine current and historical use of several substances, including on the 403 bulk compounding list, including methylcobalamin, and that's the compound and the results I will speak to today. In our study evaluating current and historical use, we first examined data coming from over 1,700 individuals with autism spectrum disorder diagnosed in the Center for Autism-Related Disorders at the Candy Krieger Institute. These individuals, upon intake at the Center for Autism and Related Disorders at Candy Krieger, were asked to complete a patient intake research questionnaire upon which a list of any medication that they had been taken or had been prescribed and taken for their autism spectrum disorder was included. Among those on the list was included B12. And notably, about 1% of individuals, or 20 of the 1,700 individuals seen at Candy Krieger, reported taking methyl B12 for their ASD-related symptoms, and all those administrations were oral. We further then linked those individual records, when possible, to the Candy Krieger pharmacy database and found that only 0.5% or 0.5% of those individuals actually had a valid prescription filled at Candy Krieger. We realized that clinical data is not really representative of the population of the true use of these substances among individuals with autism spectrum disorder, and as a result, sought to further validate and understand use of this substance in individuals with ASD for treatment specifically of symptoms related to autism spectrum disorder. So as a result, we conducted a study then using Medicaid claims files gathered from 2010 through 2014. So this included about over 369,000 individuals between the ages of 2 and 17 with autism spectrum disorder. They were all required to have an ICD-9 code of autism more than twice, or at least on 2 non-consecutive visits to confirm their diagnosis. We then linked those diagnostic codes to drug administration codes available through the National Drug Codes, as well as the Healthcare Common Procedure Coding System and the current procedural technology codes that document infusions of a substance in a medical setting. And so based on the linkages of these 3 drug mechanisms, we were able to see through the Medicaid claims data what the actual prevalence of many substances, including methyl B12, was among individuals with autism spectrum disorder over a period of 4 years across the United States. Overall, we found a prevalence of about 1% of use of methylcobalamin in this data set, and in particular relative to children with autism spectrum disorder. Notably, this prevalence did not differ from that which we saw in the general population sample from which we compared them to. Most often, injection or solution administrations were seen in the Medicaid data. However, the use of claims data, while population-based and giving us a broad sense of use across the country for treatment of autism, can help us expand upon our findings from clinical settings. We're unable at that point in time to really have a sense of what individuals' motivations for use and what particular symptoms might have been treated or perhaps perceived effectiveness. So following up to that, we conducted a population-based study working with the Simons Foundation Powering Autism Research for Knowledge, or the SPARK consortia. We conducted an online survey from December of 2019 through February of 2020, where SPARK participants and their parent or guardian were asked to respond to a series of questions regarding complementary and alternative medicines, as well as FDA-approved pharmaceuticals for a range of psychiatric disorders, including autism spectrum disorder. We had over 1,400 respondents during that short time period. Most often, the mother responded on behalf of her child with autism spectrum disorder. And overall, from this data, we found that nearly 50% of individuals had tried a complementary alternative medication for treatment of autism symptoms. Specifically in regard to methyl B12, about 6% of individuals in this data set reported they had used it, and most predominantly oral or injection routes of administration were seen. Parents reported that, or most often the mother reported that they tried or attempted B12 therapy as a result of language difficulties in their child with autism spectrum disorder, not specifically necessarily a symptom related specifically to autism-related impairment. Additionally, they report most often that there was no change in that symptomatology for which they had primarily tried the B12 medication, or that perhaps there was a slight change in that data. So based on these 3 data sources, we are led to believe that in the population overall for individuals with autism spectrum disorder, the use of methyl B12 as well as other B vitamins is relatively rare, and appears that it may have on the population level limited impact in regard to improving ASD-related symptoms. Thank you.
Thank you. We will now take remaining clarifying questions for all the methylcobalamin presenters. Please use the raise the hand icon to indicate that you have a question, and remember to put your hand down after you have asked your question. Please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. As a gentle reminder, it would be helpful to acknowledge the end of your question with a thank you and the end of your follow-up question with that is all for my questions, so we can move on to the next panel member. Do we have any questions for our presenters? Dr. Sandbrink?
Yeah, Friedhelm Sandbrink, Washington, D.C. This is a question, I think, for Dr. Fry, but also for speaker one in the public presentation. What I'm really trying to understand a little bit better, after we've all heard this really, this evidence and the many data that you presented so well about methylcobalamin, is about what about the other injectable and approved vitamin B12 preparations that we have on the market here? I know there was some reference in there, but maybe you can explain a little bit more why they may not be used or why they shouldn't be used, or what is the evidence in regard to any treatment with those products. Thank you.
Dr. Fry, if you would like to respond.
This is Dr. Day. Dr. Fry had to sign off of the meeting to go see patients.
Thank you, Dr. Day. Speaker
I'm not
number
sure if
one.
Dr. Newbrander is still available.
Yeah, Dr. Newbrander, would you be available to answer this question? Dr. Newbrander, are you on hold? Thank you. With that, we will end the time for clarifying questions and move on to the panel discussion questions. Dr. Wada, would you like to comment?
Yes, and this is, I guess, a comment before we're voting, in that it seems that part of the issues with this methylcobalamin is the number and variety of uses that have been put forward. But a lot of the discussion, at least in the last hour or so, has been on autism syndrome disorder. Is there any way to look at this as was done with oxitriptan, in that it would just be for subcutaneous use for ASD with indications that the FDA has done before?
Will the FDA, Dr. Johnson, Dr. Bermel, would you like to answer?
This is Dr. Ganley. I'm able to answer that.
Thank you, Dr. Ganley.
Yeah, so I'm not sure what the question is, because if the ingredient is placed on the 503A list, there's no restriction with regard to how it may be used. In the past, we have placed limits on the route of administration, but we're not able to restrict how a prescriber may prescribe that. So for the just taking methylcobalamin into account here, if there was a reason if you believe it should be added to the list, it have to be clear as to why you would restrict oral use versus parenteral use, for example.
Okay, so with oxitriptan, it seems you did make an exception.
The oxitriptan will be discussed this afternoon, and although the reason, I don't want to really get into a prolonged discussion of that, but the adding it, if it was added to the 503A list for that use, we recommended it only be for oral, and that's how generally it is used if you look at those reviews. And there's not evidence of it being administered parenterally, and there's obviously safety issues. That does not prevent, if it is added to the list, it does not prevent oxitriptan from being used for any condition out there.
Okay, I'm sorry, I must have been misreading your guidance that you put out for oxitriptan.
No, generally, the previous evaluation in 2015 were for different uses, and there was not evidence of effectiveness for those conditions. And so the benefit, the risk-benefit assessment was not in favor of adding it to the list. But all it really takes for the 503A list is to find a condition for which we do think there's a benefit risk assessment in favor of benefit over risk. And if it gets on the list, that doesn't prevent a prescriber to use it for anything. But we are able to limit, all we have done is limited the route of administration. For example, if it was a drug that would be topically administered, we can limit it to topical so that it would not be given orally or by parenteral administration. So those are the caveats related to this.
All right, thank you.
Thank you, Dr. Wade, for the question. We'll ask Dr. Fensky at this time to ask his question.
All right, thank you. I just need to get something clarified, maybe by the FDA. These 3 of the 4 medications that we're discussing, they have been being compounded. I think one was since 2005, another one was 2009, another one was 2011. Why is it now that these medications are just coming up for review versus being done previously? It seems to me that basically there may not be the studies out there, but I haven't really seen a lot of, especially with the methylcobalamin, a lot of adverse effects with compounding methylcobalamin. Thank you.
This is Gail Bormel. I'm not really sure I understood the question.
So Gail, what I'm asking is that so basically, if these products have been out there and been compounded since 2005, why is it just now that we're coming up with them to put on this bulk list?
This is Gail. We've only started this process since probably about 2015 to take bulk substances that were nominated for the 503A bulk list to the advisory committee. So, we had a process whereby bulk drug substances were nominated for inclusion on the 503A bulk list, and we, had those bulk substances, and we then took them and have been, since that time, reviewing them, preparing the evaluations, and taking them to the advisory committee. So, we've done a number of bulk drug substances already, and we're just now bringing 4 more. So the timing really has to do with when we started the nominations for the 503A bulk list, and then the reviews and the evaluations, and then setting up the advisory committee meetings.
Okay, that's helpful, Gail. This is my first meeting, so I just need clarification on that. That's all. Thank you. No more questions.
Thank you, Dr. Fensky. I do have a question for the FDA presenters. Could you comment on the cobalt-related side effects, the cobalt levels, et cetera, that paper that was out there based on your review? Were there any concerns? I did hear Dr. Neubrander state that it was a testing error from LabCorp that causes that to, but it's not physiologically present. But if you read that paper in detail, it is in excess of LabCorp's maximal occupational exposure levels. I'm assuming that they have accounted for that in their testing methodology, as well as they had done some in vitro studies that showed tissue damage, organ tissue damage with exposure to cobalt at those levels for neurons, etc. Would an expert on the FDA side be able to comment on that?
Yeah, this is Dr. Ganley. I'm not sure if we've seen that paper or looked at it, so I don't know if Dr. Johnson's able to answer that. She's more familiar with the data that's available.
It's May 2010 autism cohort study of cobalt levels following vitamin B12 injections. It's David Geyer, G-E-I-E-R. Published in Toxicology and Environmental Chemistry. While I recognize that seems to be a general sense that side effects with methylcobalamin are not high.
Hi, this is Sue Johnson. Unfortunately, the technical gremlins got me again and it bounced me out. We do not have that paper in our review. That's part of what we're trying to do is gather any more information that's relevant on safety and efficacy, and we will definitely look into the safety associated with cobalamin, which sounds like it could very well affect any of the cobalamins. It is not something that's in the labeling for the approved products, so we will just have to investigate it further. We appreciate you bringing it to our attention.
All right, thank you. Dr. Fensky, is your hand still raised, or do I see that you've lowered it? Have we ensured that you've had an opportunity to ask all your questions?
Yes, I forgot to lower it. Thank you.
All right. Are there any further questions? Seeing none, the committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments. We will proceed with the question to the committee and panel discussions for methylcobalamin. I would like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. Today's question is a voting question. Dr. Takia Stevenson will provide the instructions for the voting.
Question 2 is a voting question. Voting members will use the Adobe Connect platform to submit their votes for this meeting. After the chairperson has read the voting question into the record and all questions and discussion regarding the wording of the vote question are complete, the chairperson will announce that the voting will begin. If you are a voting member, you will be moved to a breakout room. A new display will appear where you can submit your vote. There will be no discussion in the breakout room. You should select the radio button, that is the round circular button in the window that corresponds to your vote, yes, no, or abstain. You should not leave the no vote choice selected. Please note that you do not need to submit or send your vote. Again, you need only to select the radio button that corresponds to your vote. You will have the opportunity to change your vote until the vote is announced as closed. Once all voting members have selected their votes, I will announce that the vote is closed. Next, the vote results will be displayed on the screen. I will read the vote results from the screen into the record. Next, the chairperson will go down the roster, and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. Are there any questions about the voting process before we begin?
I do have a question. This is Dr. Desai. I was trying to unmute, and I guess this is a question if the chair would allow. Did we already, when you were asking for committee discussion, I know that time has closed, but I was trying to unmute. Am I allowed to make a comment for the committee?
Dr. Desai, I will allow that at this point. If you have difficulty unmuting, please raise your hand. That will give me an indication that you wish to ask a question.
You're right, Dr. Galore. I did raise my hand in the chat, so thank you.
Go ahead and ask your question.
Well, my comment related to something that I don't think we've talked about as a committee, which are the number of comments that were received in the open public docket on the regulations.gov website. I was reading through all the materials prior to the meeting, and there's over 1,000 comments, many from patients and other physicians related to this issue. Obviously, there's a lot of concern about this from multiple different sides. The FDA gave us a very comprehensive presentation. Our nominators gave us a very comprehensive presentation, and then we had the OPH. My question is more procedurally, do we need to have an opportunity to discuss the public consensus that has been submitted, or is there an opportunity to discuss that now, or was this simply meant for us as committee members to review prior to the meeting and use that in our decision making?
Dr. Desai, all of this, if there's some particular points from that you would like to bring up for discussion with the panel, please do. That is what the panel discussion time is for. If there are certain comments or points that you found particularly worth sharing.
Sure. What I found, I read through these, and as I said, there was over 1,000. I'm sure all of my fellow committee members did as well as part of the pre-work. I was struck by the number of patients, particularly parents of children, who commented about their own journey with autism spectrum disease and how methylcobalamin really helped them with their process of navigating the disease and for the suffering from it. There were several also physician comments that talked about how these are used in the injectable forms in their practice, and I know that we talked about the data concern, but I still think that these public comments are very important for us to keep in mind, and I just wanted to make sure the committee was aware of that and uses it in their weighing of the decision. Thank you.
Thank you, Dr. As stated, there are 4 criteria that, as you saw at the end of the FDA discussion, that must be taken into account while making your decision: the safety, the efficacy, use, etc. And so I'm sure all of this informs the process. Did you have any other comments, Dr. Desai?
No, thank you. I just wanted to go on record as making sure that we acknowledged the comments that came in on the docket. Thank you.
Thank you, Dr. Desai. Again, it's unusual, but I do see another raised hand, Ms. Fuska Walker. Do you have a question or a comment? Ms. Fuska Walker, are you muted?
Thank you very much for turning it on. I'm having difficulty here. I tried to ask a question before we moved to this session, this part of the session. I agree with the previous speaker that I read through a lot of the comments that were made online. And I do have concerns about parents and their experience with using this product. But I had a question directly related to the product that I wanted to ask the last speaker. This product has been available for quite some time because FDA has a guidance that allows these compounds to be used until a decision is made about 503A, whether they're on the bulk list or not. And these products are being used by compounded pharmacies and physicians, and they're being distributed and used subcutaneously by injection, et cetera. I know that compounding pharmacies do not have to report adverse events. That's 503A. That's not a requirement. But are there any adverse reports being picked up over the last 16 years that this product has been used? Is anybody compiling that anywhere to see the effects of all these people that are writing online, these parents that are saying this has made such a difference for them?
Hi, this is Sue Johnson again from FDA. We talked about the FAERS and CARES data, which are our voluntary reporting systems for both dietary supplements and drugs. It's difficult to tell most of the time from the submissions what type of product has been used because the reports are generally very attenuated. But we have to surmise that from a dietary supplement oral standpoint, from an oral product standpoint, there are dietary supplements available, and that is likely the products that are used in any reports about dietary supplements or in the CARES database. FAERS database is a little bit different. Some of those could be about compounded products, but we really don't have a way to reassure ourselves of that. We do know that they're not approved products, at least in the U.S., but the U.S. number of cases was relatively small compared to the total number of cases, which include foreign data. So to answer your question, there are mechanisms where the adverse events can be reported if the investigator, clinician, or patient user chooses to. It's sometimes difficult to assess exactly what product is used. Keep in mind that each compounding pharmacy makes their own formulation, so they could differ between or among pharmacies. Not everything is a standardized product like an NDA product. And it is difficult to surmise exactly what the experience has been. We would very much like to encourage additional data that's been categorized and clarified for us to be submitted. And I know some of it has come in with the comments, and we would continue to encourage the receipt of that kind of information that helps us very much to receive voluntary reports.
Thank you very much. I have no further questions.
Thank you. Thank you, Dr. Johnson. Just as a to close out that last comment from you, just to be clear, the removal of methylcobalamin from the 503A bulks list would only prevent compounding. It would not remove access to patients with autism spectrum disorder to use oral dietary supplements, which a majority of them seem to be currently consuming. It would limit the injectable availability of methylcobalamin. Is that a reasonable assumption here?
Yes, it would.
Yes.
Sorry, that just to clarify, I'm just confirming that we are not, methylcobalamin will continue to be available in an oral formulation to anyone who wishes to use it like they do today for whichever indication under the dietary supplements. By not being on the compounding list, it would limit it for injectables, which based on the Johns Hopkins Open Public Speaker is a very small proportion of patients who use the injectable formulation.
So there were, among the nominations, a number of dosage forms proposed, including oral, nasal spray, oh goodness, I don't have the list, but there were dosage forms proposed in addition to injectable products. All compounding with methylcobalamin would cease to be allowed if methylcobalamin were not put on the 503A list. And I'm going to ask Gail Formel if she would like to add anything to that. Am I missing a perspective there?
Sure. This is Gail Formel. So if methylcobalamin is not placed on the 503A bulk list, it would not be available for compounding under 503A, period. And that is separate and apart from methylcobalamin's availability as a dietary supplement. This does not affect our vote today, and our decision today does not affect the availability of methylcobalamin on the as a dietary supplement. So, but what it would do if methylcobalamin is not placed on the 503A bulk list, that means that compounders under Section 503A, like state licensed pharmacies, would no longer be able to use methylcobalamin as a bulk drug substance to make a compounded drug product.
Thank you, Dr. Bermel. That was very helpful. I see some hands raised still for the open discussion. We'll allow a couple of minutes more, but we'll have to close this at some point. Dr. Fensky?
Thank you. I just wanted to make a comment about the reporting of adverse events. From a board of pharmacy perspective, most boards of pharmacy across the United States are actually gathering adverse effects when it comes to compounding through their board. So that may be a possibility in the future, just to be getting those adverse effects reported into some type of database. I just wanted to bring it up since we were talking about where these things were being reported, but I know boards of pharmacy are starting to collect this data. Thank you.
Thank you, Mr. Fensky. I see, Mr. Walker, that you have your hand raised. Is that because you have a further question, or have you just forgotten to retract it? Thank you. With that, we will move on. I will read the question that we will be voting into the record. FDA is proposing that not be included in the 503A bulk list.
I do apologize. This is Takia Stevenson speaking. Just before we read the question into the record, I see that Dr. Gail Bermel has a comment or question. Dr. Bermel, you can start.
Dr. Bermel.
Yes.
Thank you. I just wanted to make it clear that the there are no reporting requirements, as was mentioned before, for compounders operating under Section 503A of the Act. So even if there are adverse events that occur with some of these compounded products, even if they're reported to the boards of pharmacy, as it stands currently, there's no mandatory reporting for these comments to come into FDA. And so, you know, we always look at our FAIR system as well as our CARE system, but FAIR has to do with our drug adverse event reporting system. And that reporting system for compounded drug products is purely voluntary. So we don't really get an idea of, you know, we could never tell you the denominator of, you know, products, the compounded products made and what types of adverse events we're seeing, because it is a voluntary reporting system. It's not mandatory for these adverse events to be reported by compounders under Section 503A.
Thank you, Dr. Bermel. I will take this moment, because this has been slightly unusual, to give everybody one more chance. Is there anyone else who has a comment or a question before we move to the vote? Thank you for a robust discussion on this topic. Dr. Bermel, I still see your hand raised. Would you have another comment?
No, thank you.
Thank you, Dr. Bermel. All right, seeing as how we have concluded our discussion on this topic, I will attempt one more time to read the question into the record. FDA is proposing that methylcobalamin not be included on the 503A bulks list. Should methylcobalamin be placed on the list? Does anyone have any questions about the wording of this question? Dr. Katzman?
Yes, I just have a clarification about the wording of this. So it's like, so if, depending on how we vote, if we vote yes, that means that we're voting that we agree that it should not be included. If we vote no, then we're saying that it should be included. Is that correct?
Yes, I was going to read this part after, but I'll read it now. If you vote no, you are recommending FDA not place the bulk drug substance on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes the subject of an applicable USP or NF monograph or a component of an FDA-approved drug. Does that help answer your question?
So if I vote no, then I'm recommending it not be put on the list.
Correct.
Okay.
Does that answer all your questions, Dr. Katzman?
Yes, thank you. Thank you so much.
You're very welcome. Are there any other raised hands? Dr. Sandbrink, I don't see your hand raised at this time, though it may have been earlier. I just want to ensure we've given you an opportunity to ask a question.
Yeah, I had this is Friedhelm Tampering, Washington, D.C. I had the same clarifying question. So voting it saying it the other way around in regard to the wording. So if we vote yes, that means it will remain on the list and it becomes and remains available as it is currently.
Correct. And just for further clarification for everyone, I will repeat the clarification. If you vote no, you are recommending FDA not place the bulk drug substance on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes the subject of an applicable USP or NF monograph or a component of an FDA-approved drug. Dr. Katzman, I see your hand is still raised. Is that because you have a question? If there are no questions or comments concerning the wording of the question, we will now begin the voting on question for methylcobalamin.
We will now move voting members to the voting breakout room to vote only. There will be no discussion in the voting breakout room. Voting has closed and is now complete. Once the vote results display, I will read the vote result into the record. Voting has closed and is now complete. The vote results are displayed. I will read the vote totals into the record. The chairperson will go down the list, and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. There are 9 yeses, 5 noes, and 0 abstentions. Record. Thank you. We will now go down the list and have everyone who voted state their name and vote into the record. You may also provide justification for your vote if you wish to. We'll start with Ms. Fusco-Walker.
Yes, Sandra Fusco-Walker. I voted no based on the information provided, but I hope this compound can be addressed again with more data.
Dr. Desai.
Hi, Samal Desai. I voted yes. This was a very interesting and very dynamic discussion. I very much appreciated all of the science presented by the FDA, the nominators, and our public speakers. For me, one of the most important things remains access to care for medications for our patients, and the overwhelming number of responses in the public docket related to access of this compound is what convinced me to vote yes, in addition to the data presented during the OPH. Thank you.
Dr. McElhaney.
Linda McElhaney, I voted yes, mainly because I want these patients to have access to care so that they have quality of life. Also, it's standard of practice in 2 medical professional organizations, and so I think that we should support the experts that actually deal with these patients. Thank you.
Dr. Fensky.
Tim Fensky, I voted yes.
Dr. Rebello.
Elizabeth Rebello. I voted no based on the lack of adequate scientific evidence.
Dr. Bogner.
Robin Bogner, I voted yes based on the number and compelling stories from many of the comments, the hundreds of comments.
Dr. Katzen.
I voted yes for all the reasons stated before me, including access to care and benefits for the thousands of patients with ASD, as well as the minimal risks described today as well. Thank you.
Dr. Sun.
This is Jeannie Sun. I voted yes to support and access to these medications, and based on the testimony and in the briefing materials and the presentations that were given, and just the number of patients that are relying on this medication.
Dr. Gupta.
Thank you. I voted no. First off, I think this is a very difficult decision. I believe that although this is a very important medication, first of all, for access, I do think it's very important that we address patient safety as well. And I do think that although the presentation from the FDA was compelling, the presentation for patient access was also very compelling. And we do have to find better evidence to ensure that there is patient safety. That also is very strong. So I did vote no at this time and hope that we can overturn that decision very soon. Thank you.
Dr. Veda.
Yes, Alan Veda, I voted yes, and actually I voted yes because I wasn't convinced that there was a lot of information to show that the drug was unsafe, and it seemed that it was just because of the number of uses for that it came up. But I would like to qualify my vote also, not that it would mean anything, that it would just be for oral and subcutaneous. Thank you.
Dr. Sandbrink?
Friedhelm Sandbrink, I voted yes. I want to mention that I was left quite frustrated with the lack of comparison data between the different B12 products that are commercially available and that are FDA approved and the proposed methylcobalamin. But at the same time, I also felt that there was insufficient evidence to truly take that option away at this point.
Dr. Patel?
Yeah, hi, Clay Patel. I voted no. Agree with all of the sentiments described by the committee members here. Really difficult decision given its existing use. Though after hearing all of the various different presentations and also appreciating the importance of having the therapeutic option available, I just didn't see the level of objective clinical effectiveness that's needed to conclusively vote yes. So that said, I think one of the points that I'd like to also go back to was important from Dr. Veda with regards to how this committee gets to vote, and really would have been nice to have it be stratified where you can isolate a particular indication as opposed to broadly deciding on a bulk substance to make it available or not. Thank you.
Thank you, Dr. Patel. Padma Galore, my vote was no. I balanced similar to, as you've heard from the other panel members, clinical effectiveness. There was soft information for most of the indications for the value of methylcobalamin compounded over anything that is currently available. In specific, we discussed autism spectrum disorder in much detail, and that is actually where my concern came in. This is a population that is, sick, and they are unable to advocate for themselves. Fortunately, their parents do advocate for them. However, patient safety being where it should be, if there were side effects from this, it wouldn't be easy to discern in patients who already have complex, chronic disease. And so this more than ever is a need and a cry for science and appropriate studies. I would hope that Dr. Fry's request for funding, et cetera, though this is not the forum, be recognized, and how important it is that the people who are taking care of these patients have an opportunity to truly study this in a balanced manner. But that said, today there are risks potentially there, and while I accept that the some of the risk papers that are out there haven't been fully looked at, those were my concerns in moving this. As far as access, the fact that the oral formulation, which is what the majority of the autism spectrum disorder, including the patient comments that have come in, a majority of them seem to be using, and they would not be limited from access to that, is what influenced my vote. Thank you. We will now take a break. I just wanted to commend our panel members, the FDA, our presenters, and the public for their patience and dedication as we discuss this important topic. And it has undoubtedly delayed our lunch break. We will, given the time, only take a 20-minute break at this time, and we'll reconvene at 3 p.m. I'm sorry, is someone trying to speak?
Yes, hi, this is Kathleen Gura. You skipped over me during the roll call for the vote.
Oh, I am sincerely sorry, Dr. Gura. Let me get you back on record. Dr. Gura?
Okay, I voted yes, and the reason for my answer is if we take this option away, we're going to be forcing patients to go into the supplement world, and at that point we may not really know what the quality is of the products they're going to use to compensate for the lack of access. So I wanted to go on record for that. Thank you.
Thank you, Dr. Gura, and I apologize for having missed you in the roll call. Have we covered, Takia, have we covered everyone else?
Yes, Dr. Galore, I believe you have.
Yeah, thank you very much. As we were saying, we're going to take a break for 20 minutes and we'll reconvene at 3 p.m. Panel members, please remember that there should be no chatting or discussion of the meeting topics with other panel members. Additionally, those panel members participating in the remaining topic discussions should plan to rejoin at 3 p.m. to ensure you are connected before we reconvene. Thank you.
Hello, Dr. Galore. This is Takia Stevenson speaking. Can you hear me? Please unmute your phone if you can hear me.
Thank you, Takia. I think it had been muted from the other side. My phone was unmuted. Can you hear me now?
We sure can. Thank you so much, Dr. Galore.
Wonderful. Welcome back, everybody. Before we begin, Dr. Takia Stevenson will introduce the new special government employees, industry representatives, and FDA presenters for the afternoon topics.
Hello, this is Takia Stevenson again. Dr. Jennifer Lai, please state your name and your affiliations for the record.
Yes, hi. My name is Jennifer Lai. I'm a hepatologist at UC San Francisco and Director of Hepatology Clinical Research for UCSF Health. Thank you.
Dr. Liang Pumsakul.
Hi, my name is Dr. Sutaliang Pansko, Professor of Medicine at the Indiana University School of Medicine in Indianapolis.
Dr. Basani.
Yes, good afternoon. I'm Gus Basani. I serve as Chief Scientific Officer for PCCA in Houston, Texas.
I will now introduce the FDA participants. Dr. Wolford.
Yes, thank you. I'm Madeline Wolford. I'm a physician with the Pharmacy Compounding Review Team with the Office of New Drugs, FDA.
Dr. Hong. Dr. Hong, if you can hear me, please unmute and introduce yourself.
Sure. Hi, I'm sorry. I was double muted. This is Jay Hong. I'm with Division of Anti-Infectives, and I'm a medical officer. Thank you.
Thank you so much. I will hand it back to the chair, Dr. Galore. Thank you.
Thank you, Takia. We will now proceed with the FDA presentation on choline chloride for Dr. Suhail Qasim.
Good afternoon. I'm Suhail Qasim from the Office of New Drugs. I will discuss a nomination for choline chloride. This slide shows the review staff involved in the choline chloride evaluation. Choline chloride was proposed for use with respect to liver diseases, including non-alcoholic fatty liver disease, hepatic steatosis, fetal alcohol spectrum disorder, and atherosclerosis. In addition to these uses, this evaluation considers the use of choline chloride for supplementation in long-term total parenteral nutrition. Choline chloride has been nominated and proposed for use to be administered via the parenteral injection route. In conducting the 503A evaluation, FDA reviewed publicly available information to assess choline chloride based on 4 criteria. The first criteria applied was the information considered regarding the physical and chemical characterization of choline chloride. Choline chloride is well characterized physically and chemically and is likely to be stable under ordinary storage conditions in the proposed dosage form. I will discuss briefly the general pharmacology of choline. Choline chloride is an essential nutrient existing in many components of food, and it is designated as a generally recognized as safe nutrient in human food and animal feeds. It plays a vital role in the structural integrity of cell membranes, methylation metabolism, cholinergic neurotransmission, transmembrane signaling, lipid, and cholesterol transport. Majority of the dietary choline consumed in the United States is in the form of phosphatidylcholine. Choline is absorbed from the lumen of the small intestine, and much of its metabolism occurs in the liver, where it is found primarily as phosphatidylcholine. In addition to this dietary supply, choline is formed by de novo biosynthesis via an endogenous pathway in the liver via the sequential methylation of phosphatidyl ethanolamine using S-adenosyl methionine as the methyl donor. Next, we will discuss the safety profile of choline chloride. That's the second criteria in the 503A evaluation. I will start with the available non-clinical data. In the publicly available data, we located a risk assessment for choline chloride conducted by the EPA. The acute toxicity studies showed a large margin of safety for choline chloride in various species using various routes of administration. In an 8-month repeat dose toxicity study, an intraperitoneal injection of choline chloride in rats resulted in lung and liver abnormalities as shown on this slide at 3 months and at 6 months. These non-clinical toxicities were at much higher doses than proposed for clinical use. The standard panel of genotoxicity assays did not show genetic toxicity activity, and choline chloride did not have genotoxic adverse effects. Choline chloride is important for the normal fetal and early postnatal development. Male rats exposed to high doses of choline chloride showed a transient increase in adverse effects on spermatogonia in the first few days of the study. These non-clinical toxicities observed were at much higher doses than proposed for clinical use. No long-term carcinogenicity studies were conducted. In the FAERS database, 6 cases were identified with use of choline chloride as a weight loss injection that reported both exposure to choline chloride and at least 1 adverse event. These cases were confounded by the multiple medications in the injection or the multiple ingredient products that included choline chloride as one of the ingredients. 6 cases were reported in the CARES database that included cases when choline chloride was one of multiple ingredients in energy drinks, muscle performance products, and nutritional supplements. Insufficient information was provided to interpret the contribution of choline chloride. Considering that there were many other substances in the dietary supplement, it was not possible to determine whether a causal connection existed between choline chloride and the adverse events reported. The established adequate daily intake of choline is 550 milligrams for men and 425 milligrams for women, with an upper daily limit ranging up to 3.5 grams per day. Adverse events have been reported in clinical studies at doses much higher than the daily intake from food. Choline chloride has been associated with nausea, fishy body odor, sweating, diarrhea, and salivation. Hypotension is observed with daily use of more than 10 grams choline chloride. In most clinical studies, choline has found to be well tolerated and not associated with significant adverse events. The subsequent slides will discuss the third criteria in the 503 evaluation. And to do that, each of the uses proposed to the nomination will be discussed. Choline chloride was evaluated for the treatment of liver diseases that included the nominator's proposed use for hepatic steatosis. Hepatic steatosis, or hepatic fat accumulation, or also called fatty liver, is not specific to a disease condition. It is a histological finding with the presence of large and small vesicles of fat, predominantly triglycerides, accumulating within the hepatocytes. For instance, if one were to consume 1 or 2 cocktails or glasses of wine tonight, the next day it is quite conceivable that there will be the presence of hepatic steatosis. The causes of hepatic steatosis are shown on this slide. Typically, hepatic steatosis is not seen on imaging modality. It is usually a benign finding. Although, as we will discuss in the subsequent slides, in rare instances, over many years to several decades, chronic liver disease may develop as a result of the increased triglyceride accumulation leading to chronic inflammation, hepatocyte degeneration, and scarring, with progression to fibrosis and potentially cirrhosis. Next, we will discuss the nominators proposed use for non-alcoholic fatty liver disease, abbreviated as NAFLD. NAFLD includes a spectrum of histological changes that includes simple fatty infiltration of the liver, also known as simple or isolated steatosis, or non-alcoholic fatty liver, abbreviated as NAFL, and also non-alcoholic steatohepatitis, abbreviated as NASH, NASH, which is an extreme form of NAFLD and is regarded as a leading cause of cirrhosis in the United States. Therefore, the histological changes of hepatic steatosis are seen across the NAFLD spectrum. The figure illustrates the spectrum of NAFLD, and that hepatic steatosis is not specific to a disease condition. The histological changes are seen across the spectrum of NAFLD disease conditions. Typically, hepatic steatosis is transient and reversible. Most humans who have evidence of hepatic steatosis are asymptomatic and are not adversely affected by its presence. The figure illustrates the histological subtypes and the associated risks for progression of NAFLD to other chronic liver diseases. As shown with the blue arrow, very few patients with simple non-alcoholic fatty liver have progression to diseases like cirrhosis that is serious or life-threatening. At present, clinicians cannot predict which patients are likely to progress from benign steatosis, shown in box A, on the upper left side of the figure, to steatosis, shown in box C on the upper right side of this figure, or cirrhosis, shown in box E on the bottom of the figure. If we assume that 100 million Americans have benign steatosis or non-alcoholic fatty liver, and less than 4% wind up having cirrhosis, which is not end-stage liver disease. Non-alcoholic fatty liver is likely to be 5 times less frequent in occurrence than in patients who have non-alcoholic steatohepatitis, or abbreviated as NASH. Benign steatosis, or NAFL, is reversible. For example, by abstaining from alcoholic beverages, weight loss and exercise, improved diet, management of hypertension and dyslipidemia, and glucose control in some diabetic patients. In conclusion, hepatic steatosis is not specific to a disease condition. I will briefly discuss the relation of choline deficiency to development of hepatic steatosis and the non-alcoholic fatty liver disease spectrum. Theoretically, individuals who were to consume a diet deficient in choline can develop hepatic steatosis and go on to develop liver damage in the form of NAFLD. It is because of the lack of dietary sources of phosphatidylcholine that limits the export of excess triglycerides from the liver in lipoproteins. We discussed earlier in slide 11 about hepatic steatosis. This fat accumulation within the hepatocytes, predominantly in the form of triglycerides, can result in the non-alcoholic fatty liver disease spectrum, and that includes histological evidence of hepatic steatosis. However, since the United States diet is rich in choline-containing foods, patients are unlikely to need a change in their diet in order to include choline-rich foods, or other nutritional supplements will be available to provide that choline source. It is also known that patients on long-term total parenteral nutrition that lack adequate choline in the form of phosphatidylcholine and phosphatidylethanolamine can develop steatosis. This will be discussed further. The next few slides will discuss the evaluation of the information for effectiveness in NAFLD related to choline, including the current practice guideline recommendations. 6 practice guidelines pertaining to the treatment of NAFLD were identified. Use of choline chloride as a treatment of NAFLD was not mentioned in any of these treatment guidelines, stating that nutritional supplements do not show scientific data strong enough to support their recommendation as a treatment of NAFLD. We evaluated 3 studies in patients with NAFLD. In one trial, 43 children and adolescents aged 4 to 16 years with baseline liver biopsy confirming non-alcoholic steatohepatitis were evaluated after 6 months oral treatment with daily supplement pro-DHA steatolip plus pills containing a combination of DHA, choline, and vitamin E. All subjects were to undergo liver ultrasound and blood tests at 12 months. The primary outcome measure of improvement in liver hyperechogenicity by liver ultrasound at 12 months decreased from 50% to 5% of patients in the subgroup analysis of the active treated subjects with severe steatosis at baseline. However, the multiple ingredients in the treatment prevent attributing any benefit to choline. Observational data from a cross-section analysis of 664 subjects with biopsy-proven NAFLD who were enrolled in the multi-center prospective non-alcoholic steatohepatitis clinical research network, NASH-CRN abbreviated, was conducted to determine whether there was an association between choline intake and the histologic severity of NAFLD or NASH determined by liver biopsy. Baseline dietary data was based on recall and not prospectively collected. The study showed that postmenopausal women with a reported choline intake less than 1/2 the defined adequate intake had increased fibrosis. There was no intervention to determine whether liver disease improves with choline treatment. Another study, reported in 1946, evaluated choline for the treatment of cirrhosis of the liver with varying etiology. Due to the small number of responding patients, the multiple treatments provided, and the confusion regarding whether choline chloride was administered, it was not possible to draw any conclusion regarding the efficacy of choline chloride for the treatment of cirrhosis of the liver. FDA has, in its discretion, opted to evaluate the clinical effectiveness of choline chloride for the unnominated use of supplementation in long-term total parenteral nutrition. Choline deficiency frequently occurs in long-term total parenteral nutrition patients, and this nutritional deficiency can lead to hepatic steatosis. In 2012, the American Society for Parenteral and Enteral Nutrition published a position paper recommending that choline be routinely added to adult and pediatric parenteral nutrition formulations with even dosing guidance. However, we were not able to find data regarding the extent of use of choline chloride in parenteral nutrition, if it is in fact added to parenteral nutrition. Although the ASPEN has made this recommendation, it is not clear that it has been implemented in the treatment of patients on total parenteral nutrition. Sources of choline for parenteral nutrition are available as egg phospholipid, which is primarily phosphatidylcholine or phosphatidylethanolamine. An example of an FDA-approved product is intralipid. The body can convert each to choline through metabolic pathways. In conclusion, we could not find clinical evidence that choline administration will be effective in the treatment of NAFLD that is not related to a deficiency of choline. Next, we will discuss the evaluation of the information for effectiveness in atherosclerosis related to choline chloride. There were no scientific articles evaluating the use of choline chloride for the treatment of atherosclerosis and available in English that were cited by the nominator during the submission. The nominator included 2 references for this use that FDA did not review because the articles were in a foreign language. Although the FDA identified 3 studies evaluating the use of choline chloride in atherosclerosis. One study included a 21-day treatment with a combination of 6 nutrients administered orally, including choline, in 12 patients following acute MI. Any benefit of choline could not be determined from the multiple ingredients used. An 8-year cohort study in 16,000 women failed to show any difference in cardiovascular risk between women in the upper versus the lowest quartile of dietary choline intake. The prospective study of atherosclerosis risk and communities cohort of over 14,000 men and women found that choline intake from food was not significantly associated with incidence of coronary artery disease, and that higher choline intake was not protective for coronary heart disease. There is insufficient information to support use of choline chloride for the treatment or the prevention of atherosclerosis. Next, we will discuss the evaluation of the information for effectiveness in fetal alcohol spectrum disorder related to choline chloride. Optimal maternal nutrition is required to produce healthy offspring. When maternal nutrition status is compromised with alcohol use, essential nutrients are displaced or not obtained. Although animal models have shown that choline chloride supplementation reduces some of the detrimental effects of alcohol consumption on fetal animals, randomized controlled clinical trials evaluating the use of choline chloride to mitigate adverse effects on infant growth and cognitive function from prenatal alcohol exposure have had mixed results. Two placebo-controlled studies evaluated the developmental outcomes during the infant's first year with prenatal choline chloride exposure, following choline chloride treatment in women who reported moderate to heavy drinking during their pregnancy. We also identified two trials in which choline was evaluated in children for the treatment of fetal alcohol syndrome, that is from postnatal exposure. A prospective prevention cohort study evaluated developmental outcomes in two randomized groups of alcohol-exposed infants born to pregnant women who reported moderate to heavy drinking during their pregnancy compared to a control population of infants born to women who reported low to no drinking. Among the participants in the choline-exposed treatment arm, there was no improvement in cognitive outcome compared with the control group. And there was no difference between the group treated with only multivitamin supplement, that is group 2. The changes observed might have been solely due to the multivitamins and minerals. In an exploratory randomized double-blind placebo-controlled clinical trial, 69 heavy drinkers recruited in mid-pregnancy were randomly assigned to either 2 grams of choline or placebo. Although the authors concluded that infants born to choline-treated mothers were more likely to meet criterion on the eye blink conditioning than the placebo group, it is not clear what the changes in eye blink conditioning mean when interpreting neurodevelopmental outcome in these infants. At 12 months, the infants in the choline treatment arm had higher preferential looking at the novel stimulus on Fagan test of infant intelligence, indicating better visual memory. There were 2 trials in which choline was evaluated in children for the treatment of fetal alcohol syndrome. And this is from postnatal exposure to choline chloride. In a 6-week controlled trial in children with fetal alcohol spectrum disorders, choline or placebo was administered daily to 60 children, each aged 5 to 10 years of age. Participants in the choline group did not improve in cognitive performance in any domain compared with placebo. In a 9-month controlled trial in children with fetal alcohol spectrum disorders, choline or placebo was administered daily to 60 children aged 2.5 to 5 years of age. The study failed on the primary endpoint of global cognitive ability. We were not able to find clinical effectiveness data to support the proposed use of choline chloride with respect to liver diseases, including non-alcoholic fatty liver disease, hepatic steatosis, atherosclerosis, and fetal alcohol syndrome. There is insufficient information on the effectiveness of choline chloride for supplementation in long-term parenteral nutrition. Lastly, we apply the 4th criterion, that is the historical use of choline chloride in compounded drug products. Choline chloride has been used in pharmacy compounding since at least 1954. Based on advertising information though, choline chloride is most often used in compounded injectable products for weight loss in the United States. Choline chloride is available in the United Kingdom as part of a multiple ingredient product indicated for the prevention of vitamin deficiency in certain conditions. We have balanced the 4 criteria that was discussed to evaluate choline chloride for the 503A bulks list. After considering the information currently available, a balancing of the 4 criteria weighs against choline chloride being added to the 503A bulks list. Thank you, and that's the end of my presentation.
Thank you, Dr. Kassim. We will now take clarifying questions for FDA presenters. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and end of your follow-up question with "that is all for my questions," so we can move on to the next panel member. With that, do we have any questions? Dr. Veda?
Yes, thank you. Alan Veda. I just had the questions on the use in parenteral nutrition. It seems that you said that there's no evidence of the use in parenteral nutrition. I assume that was just looking for studies. Was Aspen ever contacted? I saw that the University of Maryland did that survey, but it didn't even look like any Aspen was even contacted in that survey. Could you answer that?
Yes, thank you for the question. So the information that I discussed were regarding the Aspen discussions on the slide was not after reaching out to Aspen, but it was based on what was reported in the literature. And I will defer to my colleagues in OCQC to add additional context regarding the University of Maryland study report.
Hi, this is Elizabeth Hankla. I can add a couple of comments about the MCRC Maryland report. So they did find evidence of compounding through the literature search, but they didn't identify anything for use in parenteral nutrition. And they did conduct a survey, which, again, they didn't find evidence of use in parenteral nutrition. I hope that helps.
Okay, but you listed the groups of associations, and Aspen wasn't one of them, or any board-certified pharmacists in parenteral nutrition? I didn't see them listed. Could you just verify that they weren't?
This is Beth again, Elizabeth Hankla. I do not see Aspen listed as associations that were contacted.
All right, thank you. No further questions.
Thank you, Dr. Veda. Dr. McElhaney?
I thought that I saw, because I wasn't sure, I hadn't heard too much about choline chloride. So I thought that I saw that it was being evaluated to become a commercial product. Or I thought I saw that it was already an orphan drug and being produced by another company. Is that true?
This is Sohail Qasim. I am not aware of an approved product that is marketed as choline chloride. And I will again defer to my colleagues in OCQC. But if anyone else has any information on the orphan product designation, but an orphan product designation does not mean that the product is marketed. It may mean that it has a certain designation provided by the FDA for product development. And I'll let others elaborate on this.
Okay, thank you. That's all I had.
Thank you. Dr. Gura?
Yes, hi. Actually, I can comment on the studied IV choline chloride injection. Back in 2018, it actually did receive orphan drug designation, and it's currently in phase 2 trials. It's almost, I think it might even be finished now. And I am a member of Aspen and board certified in nutrition, so I'm familiar with its work. There's been several centers that have been using choline in their parenteral nutrition as a means to prevent some of the hepatic complications we see with long-term PN use. But I'm looking forward to seeing the results of the phase 2 trial because it's commercial, it's a farmer-made product as opposed to a compounded product. But I just want to also find out, how does this the decisions we make today impact this phase 2 trial study drug? Are they two separate issues?
I will respond to that. This is Suhail Qasim. I will respond to that initially, and if my colleagues have additional context to add to that information. So the impact of products that are being developed for marketing purposes will not be affected based on the decisions made here. That is independent. Does that answer your question? No, sorry, Gail.
Yes, it is. Thank you very much.
And this is Gail Bormel. I was just going to echo what was just said. Yeah, they're two separate processes, and they're distinct. Have your questions been answered?
Yes, thank you.
Wonderful. Dr. Bowie?
Hi, yes, Dr. Bowie here. Dr. Qasim, you mentioned in one of your slides that choline has been used in pharmacy compounding since at least 1954. That's a long time. Do you have any information, since 1945, any other information other than related to weight loss being used in the U.S. or how it's being used in the U.K. For, I believe, for vitamin deficiencies in certain conditions? Are there any other information available regarding pharmacy compounding and maybe specifically to any safety concerns in that environment?
This is Suhail again. So my colleagues in OCQC can further elaborate on the history of use in compounding and the various uses. So the slide that was projected did specifically mention that it was in association with weight loss products. So the fares and cares cases that I discussed in the slides earlier did report adverse events. Some of them you can consider serious. However, it is not possible because in those weight loss products, either in weight loss product or when it was used for the purposes of the treating the patient, it may have been used with additional substances or the additional products. So to discern whether choline chloride was a causative agent for that adverse event, it was difficult to interpret. But what we do know is from clinical studies, there was a clinical pharmacology study that was included in the evaluation, and that included an IV injection of choline chloride, and up to 8 milligrams. I saw that there was data for up to 8 milligrams, and that was in patients with hepatic steatosis or non-alcoholic fatty liver disease. But in some patients, there needed to be downward dose titration. So that means there were adverse events noted, and those which were discussed earlier. So if that gives you the context of clinical safety from when weight loss when it is used as a weight loss product, and then regarding the products used outside the United States, well, just as you have questions, I had questions when I was looking into this issue. So I had to web search to see, are we using weight loss? How is it being used as an IV product if it is a dietary supplement? So to answer that question, I saw that there were FDA recalls on some of those products. So, and then there are some of these products are available outside the United States in Europe, which are marketed. So I cannot provide any more information regarding those products outside the United States that's available. And then I'll let my colleagues in OCQC add additional context to the historical use in compounding.
Thank you. Thank you. This is Gail Bormel. I just want to emphasize that, you know, we don't routinely get adverse event reporting for compounded drugs made by under Section 503A of the SD&C Act because it's not required. So, you know, for those, if there are adverse events, there are many adverse events associated with a particular compounded drug product that's being made under Section 503A. There's no mechanism to require reporting of those adverse events to FDA. So what we get is just, you know, a snippet of what is voluntarily reported to us. So you have to look at the adverse events under that context.
Thank you, Dr. Kasim and Gail. I have a second question. We discussed here about how choline is being used for NAFLD and NASH. Did the agency look at any investigational drugs out there being developed by other companies at this point for both NAFLD and NASH, and that may be an option for patients if choline is not available?
Before I go into discussions, this is Sohail Qasim again. My colleagues from the Division of Hepatology are available to answer questions, but before I approach them, I'm not sure whether it is a venue to, you know, discuss products under investigation, new drugs, or that are being considered in development for marketing applications in this venue. So I believe there may be, but yeah, and I leave it at that. So if my colleagues in the Division of Hepatology or other colleagues in O&D want to provide additional context to respond to Dr. Bouie's question. Thank you.
Frank Kennedy from the Division of Hepatology and Nutrition.
Hello, this is Dr. Lai, Hepatology at UC San Francisco. May I speak?
Yes, Dr. Lai, go ahead and speak.
Great. So I am one of the hepatology colleagues, and I am not aware of the use of choline chloride for the specific treatment of hepatic steatosis or non-alcoholic fatty liver disease.
Thank you.
I do have a separate question aside from that when it's my turn.
Please go ahead, Dr. Lai.
Thank you. So what we do see in hepatology is that hepatic steatosis is, in the setting of long-term parenteral nutrition, is common, and so we do see fat in the liver as a result of parenteral nutrition. And the studies that were shown by the speaker is that one of the mechanisms of this could be choline deficiency. And I wonder, I do recognize that you said that we are unaware of the frequency by which choline is added to parenteral nutrition formulations, but how would choline be added to parenteral nutrition if it is not approved today? And I wonder, if it's not approved on the bulk drugs list, is that a barrier to adding it to parenteral nutrition, and could we be actually causing harm to patients who do require long-term parenteral nutrition, particularly given the relatively mild toxicities associated with choline infusions?
Dr. Lai, I think our colleague Dr. Ananya intends to respond, but okay, go ahead, Dr. Frank.
Yeah, first of all, I wanted to concur with the statement that there is no evidence for clinical effectiveness to treat NASH or NAFLD. That was a question that came up, and I had trouble getting through because I was double muted. Regarding the addition of choline chloride to parenteral nutrition, I'm a hepatologist as well in the Division of Hepatology and Nutrition. This is a complex question, but to be brief, the use in all FDA-approved lipid supplements for parenteral nutrition include an adequate amount of phosphatidylcholine and phosphatidylethanolamine, which, as the speaker indicated, can be readily converted to choline. There are other issues, as I'm sure the hepatologist on the committee knows, that play a role in the development of parenteral and associated fatty liver disease. I think that is all I'd like to say about that. Thank you.
Thank you. That's it for my questions.
Thank you. Dr. Bowie and Dr. Gura, I do see your hands are still up. Is it because you still have questions, or?
This is Dr. Bowie. I thank you for the answers. I have no further comments. So.
Thank you, Dr. Bowie. Are there any other questions, any further questions for our presenters? Seeing none, we will now proceed with the nominator presentation. We have one presentation from Dr. Paul S. Anderson, who is speaking on behalf of American Association of Naturopathic Physicians. Dr. Anderson?
Okay, I'm assuming you can hear me?
Yes, we can.
All right, thank you. Well, thank you for this time, and thank you, FDA. That was a very excellent presentation. So I'm just going to focus on a couple of properties. So one of the things to point out is we have a lot of data on dietary requirements around all nutrients, specifically choline in this case, and we have reasonable data on the effects of deficiency in a number of areas. Now, in one of the reviews, as listed here, we have preterm birth listed there, but about 1% of all births having fetal alcohol syndrome, of which there are not many approved treatments. Also, it's pointed out that there's really no FDA-approved choline drugs with the exception of the lipid product, which has been mentioned already. And so the addition and the need for choline products for physicians through compounding is necessary. Overall, in preterm infants, choline deficiency has been noted, and it's also been noted that breastfeeding does not supply enough choline to correct said deficiencies. And this would also apply to other infants, including those affected by fetal alcohol syndrome. And we have some other just general information here that is related to deficiency, especially in preterm infants. Now, and in my presentation, I've used endnotes, and because of the way that the screen is, I don't have access to the endnotes, but we have the references cited there. Generally speaking, in babies with fetal alcohol syndrome, their choline supplementation was well tolerated, very low amounts of adverse events. And I believe in the prior presentation, cognitive characteristic changes were shown. What I would say is in particular in reference number 8, there were positive cognitive changes noted in the fetal alcohol syndrome babies. To reiterate, the supplementation of choline is beneficial for children during developmental stages, especially with respect to neurological development. And because right now the only FDA-approved treatment is a parenteral treatment, there isn't anything in the 503 setting that would be available to be used for an infant for supplementation. The other data that is out shows that both in animals and humans there are improved pregnancy outcomes with choline supplementation prenatally. And as this particular citation notes, most pregnant women in the U.S. are not achieving choline intake recommendations of 450 milligrams a day. And so while it is certainly available through the diet, it is not being generally achieved in pregnancy. And also, although this is not one of the nominated areas, there are, as most know, an increase in neural tube defects in women achieving less than 300 milligrams choline, which does fall into the previous slide as well. And I think that this was well covered by the FDA presentation. It's officially recognized since 1998. And although it's been traditionally used in compounding since the 1950s at the very least. In a different citation, number 13 at the end, it's noted that choline intakes in people other than in pregnant women, but including pregnant women, are below adequate intake levels established by IOM. Now, aspen's been brought up a few times, and this is essentially, I think, the same information that was given in the FDA presentation. So aspen is calling for a parenteral choline product to be developed and available. I do not have a slide on it, but I wanted to mention as a follow-up to some previous discussion that we just had that there is a choline chloride intravenous product that is 1 year ago, 5/26/2020, was granted a fast track by FDA for the indication of intestinal failure associated liver disease, IFALD, and that is a choline chloride IV product specifically developed by Protara Therapeutics. So it is, I believe, now in phase 3 trials. So while the parenteral choline product is hopefully going to be available, in the meantime, a choline chloride parenteral product is not available outside of its donor products in the lipid emulsions, etc. So in this particular presentation, I wanted to focus on fetal alcohol syndrome children, the potential for use in prenatal, and then the availability through 5038 compounding for a choline chloride intravenous product, because even though there's a fast track for a manufactured product, it is not currently available to the degree that I can find in any FDA information anyway. One reason for compounding that is important is there are times when people are incompatible due to allergy or sensitivity with the currently approved products. And as has been mentioned a couple of times in previous discussion and the FDA presentation, we have a parenteral source as a phospholipid approved product, and that one was mentioned a few times. In the case of compounding, if you want a choline donor that is not from an egg phospholipid source, say somebody has egg allergy, which there are data that does exist in humans, choline chloride is the preferred option for a non-egg based choline product. And so in the case of a an 503 application, choline chloride would leave an inclusion that did not cross over with any sensitivities for the egg base. Those are the references. So I will yield now for the Q&A.
Thank you. We will now take clarifying questions for the nominated presenter. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and end of your follow-up question with "that is all for my questions," so we can move on to the next panel member. With that, do we have any questions for our nominated presenters? Dr. Ganley, I see that your hand is raised. Would you have a question for our nominated presenter?
Yes, for Dr. Anderson. I guess I'm trying to understand here from your presentation, you seem to have different buckets of things. One is nutritional supplementation, which presumably could be done orally and for which there's dietary supplements available. But then you're getting into the treatment of diseases for which we have not had any evidence of effectiveness that we were able to find for the nominated conditions. And so I guess I'm wondering where the line is drawn between something that would just go on the 503A list to treat conditions for which we don't know that they would actually work versus conducting studies under a investigational new drug application to determine effectiveness.
Yes, I think that I think you make a reasonable point. There certainly is, and I think that could be said for a lot of the 503A products. There's a lot of crossover with, say, nutritional supplement availability. One of the things in regard to the bucket, as you mentioned, of nutritional use or nutritional supplement use in say for preventing deficiency as an example, with 503A is there are in many cases the need for 503A to compound a nutritional product that may include something such as, say, choline chloride and also potentially other components of the nutritional augmentation. So if choline chloride were available as a bulk substance for 503A and the physician needed that part of the compound, then the compound that was being ordered would be obviously deficient in that area. And like in the discussion of, say, methylcobalamin, there is oral availability through the supplement world. It would divorce the ability of the 503A pharmacy to be able to use that in the compound, as it were. And with respect to the disease states, the disease state I focused on was fetal alcohol, and I think that, and I cannot retrieve anything on my screen and leave the FDA portal up, so I don't have access to those notes. But the data is, as many things are, not extremely conclusive with respect to fetal alcohol, but there is positive data that I see there. So that's why I focused on that particular disease.
Yeah, just to follow up on that, I'm not sure I saw the data for the effectiveness in treating fetal alcohol syndrome, and I think the issue comes down to using a treatment, particularly in very young children, for which we don't have evidence that actually provides benefit. And so I'm trying to understand, well, why would we do that under 503A versus actually getting it done under an IND where there's where there does have some benefit in that population of patients.
No, that's a very good point with respect to, especially with respect to fetal alcohol children. And it certainly would be, you know, exactly the same as the methyl B12 and the, say, autism discussion. That certainly is the, would be the preferred and the safest pathway. It's used appropriately in adults and children. Choline is overwhelmingly safe with very low adverse events. So you, much like in the B12 discussion, you could still have it available in 503A for adults and children and still pursue it through the IND process.
Did that answer your question?
Yes.
Thank you. Do we have anyone else who has a question? Seeing as there are none, thank you very much for your nominator presentation. We appreciate it. We will now begin the open public hearing session. I would like to state into the record that there are no open public hearing speakers for this topic. The open public hearing portion of this meeting has now concluded, and we will no longer take comments from the audience. We have an opportunity now for clarifying questions. We will now take remaining clarifying questions for all the choline chloride presenters. Please use the raise hand icon to indicate that you have a question, and remember to put your hand down after you have asked your question. Please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. As a gentle reminder, it would be helpful to acknowledge the end of your question with a thank you, and end of your follow-up question with that is all for my questions. So we can move on to the next one. With that, do we have any questions for clarification? Seeing that there are no clarifying questions for our presenters, the panel discussion will now start. We encourage all our panel members to use this opportunity to bring up comments or summarize any concerns. Dr. Wada?
Yes, I just had a question probably from two of the panel members that I think it was Dr. Gura that mentioned that the choline chloride is available for parenteral nutrition right now in a study. Now, does that mean that it is that anyone could get that right now?
No, that's an investigational product. Oh, I'm sorry.
Okay. Thank you.
Does that answer your question, Dr. Maida?
Yes, it does. Thank you.
No further questions, I'm assuming. Dr. Bogner?
Thank you. I find the choline literature very difficult because people refer to lots of different cholines as choline, whereas free choline is also choline, in addition to the choline chloride, of course, which is also free choline. And I'm trying to determine, and I'm wondering if any of the panel members can clarify, or even perhaps Dr. Gora, who's obviously an expert in this field, what is the advantage of choline chloride versus choline in some sort of phosphatidylcholine? Is there an advantage or disadvantage?
Well, I don't consider my...
Thank you.
Oh, it's okay for me to answer?
Yes, please speak.
Okay. I'm not an expert in choline. I'm familiar with some of the literature that it's been used for the past several decades, I think over 20 years now, in attempts to mitigate the hepatotoxicity of parenteral nutrition. I think the thought is inadequate amounts of choline available in the phosphatidylcholine emulsifying, in the emulsifier used in the IV lipid emulsions. Not all patients receive lipid emulsions in adequate amounts. Some only get enough to prevent essential fatty acid disease. So that it might be one of the considerations. I know in my practice we don't really consider the amount of choline present in the lipid emulsions. And as I mentioned previously, I'm more intrigued with learning about the results from that the recent trials with the parenteral choline chloride that's underway, I guess now in phase 3.
Thank you, Dr. Gora. I'm wondering if anyone else has anything to add. And if not, thank you all.
Hello, I had my hand raised. This is Kuldeep.
Yes, Dr. Patel, I think that was just Dr. Bogner saying that she was done with her questions.
Okay.
Dr. Patel, please ask yours.
Yeah, thank you. To Dr. Gora's point, it seemed like based on Dr. Anderson's presentation that a subset of populations of infants or children who may potentially have an egg allergy who are on long-term TPN would likely be running into a situation of not having a therapeutic option available if the committee decides to not add it to the bulk list. So I'm wondering if there's any data on that subset of population who might have either had suffered adverse events or maybe negative health outcomes as a result of not having that option available. So I'm just wondering if there's any case reports or any data that might be out there with regards to those patients suffering from such an issue. End of my question.
I'm not aware of any, except for what we've seen in the adult literature, not in the pediatric literature, at least in regards to parenterally fed children.
Thank you. Dr. Patel, does that answer your question?
It does. Thank you.
Is there anyone else who has any comments or questions for this phase? Seeing none, the committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee as well as the public comments. We will proceed with the question to the committee and panel discussions for choline chloride. I would like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. Today's question is a voting question. Dr. Takia Stevenson will provide the instructions for the voting.
Question 3 is a voting question. Voting members will use the Adobe Connect platform to submit their votes for this meeting. After the chairperson has read the voting question into the record and all questions and discussion regarding the wording of the vote question are complete, the chairperson will announce the voting will begin. If you are a voting member, you will be moved to a breakout room. A new display will appear where you can submit your vote. There will be no discussion in the breakout room. You should select the ready button, that is the round circular button in the window that corresponds to your vote, yes, no, or abstain. You should not leave the no vote choice selected. Please note that you do not need to submit or send your vote. Again, you need only to select the radio button that corresponds to your vote. You will have the opportunity to change your vote until the vote is announced as closed. Once all voting members have selected their vote, I will announce that the vote is closed. Next, the vote results will be displayed on the screen. I will read the vote results from the screen into the record. Next, the chairperson will go down the roster and each voting member will state their name and their vote into the record. You can also state the reasons why you voted as you did if you want to. Are there any questions about the voting process before we begin?
Dr. Patel, do you still have a question?
No, I do not. I'm sorry. I'll take the handout.
No worries. Thank you. Seeing as there are no clarifying questions on the voting process, I will now read the question at hand into the record. FDA is proposing that choline chloride not be included on the 503A bulks list. Should choline chloride be placed on the list? Do panel members have any questions or issues with the wording? To clarify, if you vote no, you are recommending FDA not place the bulk drug substance on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may not use the drug for compounding under Section 503A unless it becomes the subject of an applicable USB or NF monograph or a component of an FDA-approved drug. If there are no questions or comments concerning the wording of the question, we will now begin the voting on question for choline chloride.
We will now move voting members to the voting breakout room to vote only. There will be no discussion in the voting breakout room. Voting has closed and is now complete. Once the vote results display, I will read the vote result into the record. Voting has closed and is now complete. The votes are displayed. I will read the vote totals into the record. The chairperson will go down a list and each voting member will state their name and their vote into the record. You can also state the reasons why you voted as you did if you want to. There are 2 yeses, 11 noes, and 0 abstentions. Sorry, 1 abstention. My mistake, 1 abstention.
Thank you, Takia. To confirm, 2 yeses, 11 noes, and 1 abstain. Correct?
Correct, yes. Thank you.
We will now go down the list and have everyone who voted state their name and vote into the record. You may also provide justification for your vote if you wish to. We will start with Dr. McElhaney.
Linda McElhaney, I vote no.
Dr. Fensky.
Tim Fensky, I voted no.
Padma Galore, I voted no. Dr. Gura.
Kathleen Gura, I vote no.
Dr. Ribello.
Elizabeth Ribello, I voted no.
Ms. Fusco-Walker.
Sandra Fusco-Walker, I abstained due to technical difficulties. I could not hear the presentation.
I'm sorry to hear that, Ms. Fusco-Walker. Dr. Lay.
Jennifer Lai, I vote no.
Dr. Liang Punsaku.
Suthaliang Pansu, I vote no.
Dr. Bogner.
Robin Bogner, I voted yes. I didn't feel like there was very good information either way, and don't agree with some of what was said previously, that people wish that we could revisit things in the future again. I find they don't get revisited very quickly, so I went on the side of access.
Thank you, Dr. Bogner. Dr. Gupta.
Hi, thank you. I voted yes. Similarly, I believe in the situation for patient access, based on the discussion about fetal alcohol syndrome and also the pervasiveness of this condition and also the need for access in this condition, I felt a compelling reason to vote yes. So thank you.
Thank you, Dr. Gupta. Dr. Sun.
This is Janie Sun. I voted no.
Dr. Veda.
Alan Veda, I voted no.
Dr. Patel.
Kuldeep Patel, I voted no.
Dr. Desai.
Samal Desai, I voted no.
Thank you. Takia, can we confirm that we recorded everyone's vote?
Yes, I confirm we've recorded everyone's vote. Thank you so much, Dr. Galore.
Thank you. With this, we end the choline chloride discussion. Thank you, everyone. We will now take a break until 4:30. We will reconvene, as I just stated, at 4:30 Eastern Time for the oxitriptan topic. Panel members, please remember that there should be no chatting or discussion of the meeting topics with other panel members during the break. Thank you. Welcome back, everyone. We will now proceed with the FDA presentation on oxitriptan from Dr. Madeline Wolfert.
Thank you so much. Good afternoon. My name is Madeline Wolfert. I am a physician with the Pharmacy Compounding Review Team in the Office of New Drugs, and I will be discussing oxitriptan, also known as 5-hydroxytryptophan or 5-HTP. I would like to recognize the entire evaluation team, including Drs. Lopez and Henkla, as well as acknowledge the contribution of many other FDA colleagues who helped in this effort. Our special thanks to Division of Rare Diseases and Medical Genetics in the Office of New Drugs. As I'll discuss later in this presentation, we did not perform a full chemistry and non-clinical evaluation for this review, but I'd like to recognize and appreciate the presence of our colleagues from chemistry and non-clinical disciplines who are present in this meeting to help address any relevant concerns should they come up. Oxitriptan, again also known as 5-hydroxytryptophan or 5-HTP, is being considered for inclusion on the list of bulk drug substances for use in compounding under Section 503A of the FDA&C Act, also known as the 503A bulks list. Oxitriptan is proposed for oral use in the treatment of the rare disease tetrahydrobiopterin or BH4 deficiency. The next two slides offer some background on the regulatory timeline of compounded oxitriptan. In June 2015, oxitriptan was discussed at the PCAC meeting. Oxitriptan was previously nominated for use in the treatment of depression and sleep disorders. FDA evaluated oxitriptan for inclusion on the 503A bulks list for the nominated conditions, depression and sleep disorders. Treatment for BH4 deficiency was not considered as it was not nominated for this condition. FDA did not recommend inclusion on the list due to a lack of adequate evidence of effectiveness, as well as safety concerns, including risk for serotonin syndrome and inadequately treated depression. Based on the information presented, the PCAC voted not to include oxitriptan on the list, with 10 no's and 2 yeses. Taking into consideration the PCAC's advice, FDA determined not to include oxitriptan on the list. In December 2016, FDA published a proposed rule to not include oxitriptan on the 503A bulks list. FDA received comments on the proposed rule, but none identified treatment of BH4 deficiency as a proposed use of oxitriptan. In February 2019, the final rule was published in which oxitriptan was not included on the list. Thereafter, several healthcare providers and caregivers of patients with BH4 deficiency contacted FDA, expressing that oxitriptan is an essential and standard treatment for BH4 deficiency. FDA received a citizen petition communicating that compounded drug products containing oxitriptan are used to treat patients with BH4 deficiency. In July 2019, in light of the information brought to our attention, FDA issued guidance that it generally does not intend to take action against compounders who use oral oxitriptan to compound for identified individual patients with BH4 deficiency. And now we are reevaluating whether oxitriptan should be added to the 503A bulks list for BH4 deficiency. This slide lists the criteria we consider when conducting evaluations for the 503A bulks list. These include the physical and chemical characterization, clinical and non-clinical safety, available evidence of effectiveness or lack of effectiveness, and historical use in compounding. The FDA's 2015 evaluation of oxitriptan reviewed physical and chemical characterization and non-clinical safety. Thus, full chemistry and non-clinical assessments were not performed again for our current evaluation. Today's presentation focuses on application of these criteria to oxitriptan in the treatment of BH4 deficiency. As just mentioned, the physical and chemical characterization of oxitriptan was evaluated in 2015, and the following is the summary. Oxitriptan is a relatively simple, well-characterized active pharmaceutical ingredient. It is likely to be stable in solid and solution formulations. It is unlikely to contain significant amounts of toxic impurities. From a chemistry perspective, oxitriptan appears to be acceptable for inclusion on the list. Non-clinical safety data for oxitriptan was also evaluated in 2015. The available non-clinical data on oxitriptan did not identify any particular safety concerns. There is no publicly available information on carcinogenic potential and minimal available data on general toxicity and mutagenicity. In the next few slides, I'll be presenting clinical safety information on oxitriptan. Consistent with FDA's 2015 evaluation, common adverse reactions of oxitriptan include gastrointestinal symptoms such as nausea, vomiting, diarrhea, as well as headache and dizziness. There is also the potential risk of serotonin syndrome. Serotonin syndrome is a clinically diagnosed condition that occurs with hyperstimulation of serotonin receptors in the body. It presents with a variety of symptoms that may include restlessness, confusion, shivering, tachycardia, hypertension, diarrhea, muscle twitches, hyperthermia, seizures, loss of consciousness, or even death. Based on the mechanism of action as a chemical precursor in the biosynthesis of serotonin, concomitant use of oxitriptan with antidepressant drugs could result in serotonin syndrome. In our safety evaluation, we also searched for reports listing oxitriptan as an ingredient in an adverse event report. CIFSAN collects reports of adverse events involving food, cosmetics, and dietary supplements in the CIFSAN Adverse Event Reporting System, or CARES. A search of CARES was conducted in July 2018, with an interim follow-up search in March 2021 for reports listing oxitriptan as an ingredient in an adverse event report. A total of 249 reports were identified. Most of the reports involve an oxitriptan product formulated with multiple other substances or concomitant use of other products. There is also wide variability in the quality of the reports, and some provide sparse or confusing descriptions. This limits determination of a causal relationship between oxitriptan and the adverse event reported. The database does have reports for oxitriptan identifying possible serotonin syndrome. An example of reported serotonin syndrome in the CARES database describes a 35-year-old female taking an unspecified dose of oxitriptan and Pharmagaba 250, a dietary supplement containing gamma-aminobutyric acid, 250 milligrams. She developed palpitations, elevated blood pressure, and bilateral cramping of her calf muscles. She was diagnosed with serotonin syndrome after a cardiac workup was negative, and she had an elevated serotonin level on lab testing. 2 months later, after stopping supplements, the serotonin level was within reference range, and she was asymptomatic and feeling back to normal. No clinical trials assessing safety of oxitriptan in patients with BH4 deficiency were identified. Specifically in patients with BH4 deficiency, the most common adverse effects reported for oxitriptan were gastrointestinal symptoms such as nausea, vomiting, diarrhea, and abdominal pain. Irritability, motor disorders such as chorioathetoid, dyskinetic, or myoclonic movements, and sweating were also reported. In most cases, oxitriptan is administered in combination with other medications and interventions to treat BH4 deficiency, such as L-DOPA in combination with a decarboxylase inhibitor like carbidopa, BH4 supplementation with a synthetic analog such as sapropterin dihydrochloride, and or a reduced phenylalanine diet. This could confound interpretation of adverse effects. Due to these safety risks I've discussed, specifically serotonin syndrome associated with oxitriptan use, if FDA places oxitriptan on the 503A bulks list, FDA intends to make safety information about the use of oxitriptan available to prescribers, pharmacists, and the public through information on FDA's website, in a safety guide, or through other appropriate mechanisms. I will now switch gears to talk about some background on BH4 deficiency, the role of BH4 in the body, and briefly describe the disease and its treatment. BH4 deficiency is a general term for a group of inborn errors of metabolism characterized by deficiency of the cofactor BH4. I apologize for the busy slide, but it helps to illustrate the role of BH4 in these pathways. In this figure, BH4 is highlighted in red. BH4 is an essential cofactor for multiple enzymes, including phenylalanine hydroxylase, tyrosine hydroxylase, and tryptophan hydroxylase. A cofactor is a substance in the body that is necessary for the proper function of certain enzymes. Tyrosine hydroxylase and tryptophan hydroxylase are key enzymes involved in the conversion of precursors L-DOPA and oxitriptan to critical neurotransmitters dopamine and serotonin respectively. Oxitriptan is denoted as 5-OH-TRP in this figure and is highlighted in yellow. BH4 deficiency disease is caused by pathogenic mutations in the genes encoding the enzymes in its biosynthesis or regeneration pathways, resulting in a depletion of available BH4. Thus, BH4 deficiency really comprises a group of heterogeneous neurotransmitter disorders. Deficiency of BH4 limits its availability as a cofactor, and thereby BH4 deficiency typically manifests with hyperphenylalaninemia and deficiency of the neurotransmitter precursors L-DOPA and oxitriptan. Diagnosis is usually made based on elevated phenylalanine levels detected on the neonatal phenylketonuria or PKU screen. Workup typically includes analysis of blood and urine, cerebrospinal fluid, and gene sequencing. BH4 deficiency disorders are commonly characterized by motor dysfunction, impaired muscle tone, movement abnormalities, intellectual disability, and seizures. Symptoms of BH4 deficiency typically present in infancy, such as in the first few weeks of life with poor suck and decreased spontaneous movements, but are often noted around 4 months of age. BH4 deficiency is a rare disease, estimated to affect approximately 1 in 1 million individuals in the general population. The exact prevalence is difficult to determine given variability in newborn screening protocols, and that some cases, especially if mild, may go undiagnosed or misdiagnosed. The treatment strategy described in the literature is two-pronged. First, limitation of hyperphenylalaninemia through a restricted phenylalanine diet and or BH4 replacement. And secondly, in combination with phenylalanine control, substitution of depleted neurotransmitters by using the precursors oral oxitriptan and L-DOPA carbidopa. We were unable to find information on alternate routes of administration, so our evaluation is limited to oral administration of these medications. Treatment should be initiated as early as possible to optimize neurodevelopmental outcome and improve and prevent worsening of symptoms. Late detection and late initiation of effective treatment can lead to irreversible brain damage. Treatment is focused on managing the symptoms and preventing long-term nervous system damage. Patients with BH4 deficiency require close and lifelong follow-up. Pediatric patients require frequent visits due to dosing titration and dose adjustments with weight gain. In the next slides, I'll present the information we found on the effectiveness of oxitriptan for BH4 deficiency and some case reports. Oxitriptan is considered first-line treatment in BH4 deficiency in the literature. A consensus guideline for the diagnosis and treatment of BH4 deficiencies published in 2020 included a strong recommendation that from a biochemical standpoint, oxitriptan is considered first-line treatment as benefits clearly outweigh adverse effects. NORD, the National Organization for Rare Disorders, identifies oxitriptan as a standard therapy used to restore neurotransmitter balance. The recommended pediatric target dose for oxitriptan is variable in the literature, but has been cited as 4 to 10 milligrams per kilogram per day orally. The starting dose is 1 to 2 mg per kg per day, typically divided in 3 to 6 doses per day. This is slowly titrated up based on clinical response, side effects, and weight gain in pediatric patients. Measurement of neurotransmitter metabolites in cerebrospinal fluid may also be helpful in dose titration. Numerous case reports worldwide regarding treatment for BH4 deficiency with oxitriptan have been published. Some case report examples in children include an infant showed steady improvement in myoclonus, uncontrolled movements, hypersalivation, and head control when treated with oxitriptan and L-DOPA. A 27-month-old child showed improvement in areas of receptive language and motor strength, in addition to increased alertness and responsiveness after treatment with L-DOPA, carbidopa, and oxitriptan. A long-term follow-up report of 5 patients with BH4 deficiency shows the range of disease severity and variation in treatment response. Some patients demonstrated improvement in symptoms such as swallowing difficulties and seizures with oxitriptan and L-dopa-carbidopa. Here's what we found on the historical use of oxitriptan in compounding. Based on published literature, oxitriptan has been used in pharmacy compounding for BH4 deficiency since at least 2011. However, it can be presumed that it's been used since the 1970s when dosing for children with BH4 deficiency was first described. So we have revisited whether oxitriptan should be added to the 503A bulks list, addressing the physical and chemical characteristics, safety, effectiveness, and historical use and compounding of oxitriptan for the treatment of BH4 deficiency. Based on this information, the agency has considered a balancing of the criteria weighs in favor of oxitriptan for oral administration being added to the 503A bulks list. Thank you very much. This concludes my presentation.
Thank you for that presentation. We will now take clarifying questions for FDA presenters. Please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you've asked your question. When acknowledged, please remember to state your name for the record before you speak and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you and the end of your follow-up question. With that is all for my questions so that we can move on to the next panel member. With that, do we have any questions? Dr. Veda?
Yes, thank you. I just want to clarify. As of right now, oxitriptan isn't on the list, but the FDA has a guidance that says that it can be used for oral compounding for BH4 deficiency with restrictions. Is that correct?
Yes, that's correct. So, sorry, can I answer this, Matt, a little?
Yes. Yes, please.
It was voted in 2015 not to be included on the list, and it was subsequently not included on the list. The guidance allowed for compounding with those certain restrictions, oral administration, and for patients with BH4 deficiency, with the intention to reevaluate it for inclusion on the 503A bulks list.
Okay, just to follow up then, if we do include it on the list, then it once again can be used for insomnia and depression with the adverse events that have been identified. Is that correct?
So including it on the list would not limit the indication, yes. And I can also refer this question to Gail Bormel and OCQC for additional information as well.
Yes, what was said, this is Gail Bormel, what was said is correct. So Dr. Veda, when we put out that guidance, it was sort of a policy to tell people when we would not object to the compounding of drug products using oxytryptan because we had actually a final rule which said that oxytryptan would not be placed on the bulk list. So because we found out about this use for oxytryptan after the final rule published, we we issued the guidance so that 503A compounders could still use it, within the limits described in the guidance. What we're now doing is revisiting whether oxytryptan for oral administration should be placed on the 503A bulks list. And it's correct that when we place oxytryptan on the list, we cannot limit the indication or how the bulk drug substance, what the bulk drug substance that made into a compounded drug product would be used for.
Okay, I'm sorry, just so one other follow-up then is that is there any in between that we could just stay with what's current? That it's not on the list?
The guidance, just to retain the final guidance?
Correct.
You know, it's possible that we would not have to alter the way things are right now, but remember, we have under 503A there is a scheme that that is described in order to tell compounders under Section 503A what types of bulk drug substances they can be used in compounding, and that those bulk drug substances that can be used are those that are components of FDA-approved drugs, those that are subject to an applicable USPRNF monograph, or those that appear on the 503A bulk drug list. There's no mention in the law of, a discretion policy and guidances that would, implement the list. So because we had new information, about oxitriptan, we brought it back to the expert, the 503A the Pharmacy Compounding Advisory Committee, to reconsider.
Okay, no further questions. Thank you.
Thank you, Dr. Veda. Dr. Barmel, if I could follow up on Dr. Veda's question, just to elaborate, just for some elaboration and clarification. So the FDA recommendation is, the presentation seems to be for a specific indication, but the side effects for other indications which were a concern to start still exist. Is the recommendation to include this in spite of those potential side effects?
I think that definitely the reason we're looking at this bulk drug substances because we found out about the use of oxitriptan in compounded drugs, for BH4 deficiency. So once as is the case when we do add bulk drug substances to the 503A list, we can't limit it by indication. We can't limit it by uses. And, there may be some uses that cause adverse events. It's possible. But we have to, we know that there are uses for which this bulk drug substance are needed for certain other uses. And so, we put it on, we may limit it to the route of administration, but we recommend something go on a list, for what it's being evaluated for, and we do not, we don't limit it for other particular uses because we don't have the ability to do so. We'd have to rely on the provider who's writing a, patient-specific prescription for the, the patient to, write the prescription for the use and to appropriately monitor the patient.
Thank you, Dr. Barmel. Dr. Bowie?
Yes, Dr. Bowie here. Question, Dr. Wolford. First, my comments. I commend the agencies for the commitments to make the safety information available to the public, and that's very helpful to the public. My question is relating to pediatric patients. You mentioned dosing. What's the lowest age group that oxitriptan can be used for pediatric patients?
So, the evaluation for these patients begins with, let's say an abnormal newborn screen at birth. So once the deficiency BH4 deficiency is identified and a concern for neurotransmitter depletion or derangement has been identified, my understanding is it would be started as early as possible. There is recommended dosing in the literature in the newborn age group. That might be a starting dosage of 1 to 2 mg per kg per day, which would be slowly titrated up. But it is indicated in that age group once diagnosed.
And I can certainly also refer that question to our colleagues in Division of Medical Genetics and Rare Diseases too, if they have anything else to add.
Ms. Hyde, can you hear me?
Yes.
Yeah, I don't have anything else to add. I completely agree with the answer. This is Dr. Zboku from our Rare Diseases team. Thanks.
Thank you. No more questions from me.
Thank you. Do we have any other questions for our presenters? Seeing as there are none, we will be opening the open public hearing session. I would like to state into the record that there are no open public hearing speakers for this topic. The open public hearing portion of this meeting has now concluded, and we will no longer take comments from the audience. Are there any further clarifying questions for the presenters? Or we will consider this the start of the panel discussion. And we are open to comments from panel members as well. Dr. Veda?
Yes, just one other question. Is there any data from like 2019 until current that it's been used under the new guidance?
This is Gail Bormel. I believe that you know, the data we have is really the calls that we received and the documents that we received, letting us know that this was important to be used in compounding for this particular use, and that it needed to continue. So I don't, that's all the data that we have, that the guidance was necessary, and you know, has it has been, you know, well received. But it's, you know, it's anecdotal.
Okay, thank you. No further questions.
We do not, just so you know, Dr. Veda, we don't receive information on what pharmacies are making, what state licensed pharmacies, what federal facilities, what you know, licensed physicians are compounding under Section 503A. There's no way for that information to be reported to us. You know, currently we just don't receive that.
Okay, thank you.
Thank you. Are there any other comments or discussion points? Seeing as there are none, the committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments. We will proceed with the question to the committee and panel discussions. I would like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. Today's question is a voting question. Dr. Takia Stevenson will provide the instructions for the voting.
Question 4 is a voting question. Voting members will use the Adobe Connect platform to submit their votes for this meeting. After the chairperson has read the voting question into the record and all questions in discussion regarding the wording of the vote question are complete, the chairperson will announce that the voting will begin. If you are a voting member, you will be moved to a breakout room. A new display will appear where you can submit your vote. There will be no discussion in the breakout room. You should select the radio button, that is the round circular button in the window that corresponds to your vote, yes, no, or abstain. You should not leave the no vote choice selected. Please note that you do not need to submit or send your vote. Again, you need only to select the radio button that corresponds to your vote. You will have the opportunity to change your vote until the vote is announced as closed. Once all voting members have selected their vote, I will announce that the vote is closed. Next, the vote results will be displayed on the screen. I will read the vote results from the screen into the record. Next, the chairperson will go down the roster and each voting member will state their name and their vote into the record. You can also state the reasons why you voted as you did if you want to. Are there any questions about the voting process before we begin?
Seeing as there are no questions, I will now read the question into the record. FDA is proposing that oxitriptan for oral administration be included on the 503A bulks list. Should oxitriptan for oral administration be placed on the list? Any questions or concerns with the wording of this question? I do not see any raised hands. We'll clarify that if you vote no, you are recommending FDA not place the bulk drug substance on the 503A bulks list. If the substance is not on the list when the final rule is promulgated, compounders may use the drug for compounding under Section 503A unless it becomes the subject of an applicable USP or NF monograph or a component of an FDA-approved drug. If there are no questions or comments concerning the wording of the question, we will now begin the voting on the question for oxitriptan.
We will now move voting members to the voting breakout room to vote only. There will be no discussion in the voting breakout room. Voting has closed and is now complete. Once the vote results display, I will read the vote result into the record. Voting has closed and is now complete. The vote results are displayed. I will read the vote totals into the record. The chairperson will go down a list and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. There are 11 yeses, 0 noes, 1 abstention.
Thank you. We will now go down the list and have everyone who voted state their name and vote into the record. You may also provide justification for your vote if you wish to. We will start with Dr. McElhaney.
Linda McElhaney, I vote yes.
Dr. Desai.
Kamal Desai, I experienced technical difficulty during the voting and was not able to vote as a panel member, therefore had to abstain.
Sorry to hear that, Dr. Desai. Dr. Fensky.
Tim Fensky, I voted yes.
Padma Galore, I voted yes. Dr. Rubello.
Elizabeth Rubello, I voted yes.
Dr. Bogner.
Robin Bogner, I voted yes, and I'd like to say I stand corrected. We do revisit the bulk list when things are not accepted the first time. Thank you.
Thank you, Dr. Wagner. Dr. Gura?
Kathleen Gura, I voted yes.
Dr. Sun?
Jeannie Sun, I voted yes.
Dr. Veda?
Yes, Alan Veda, and I voted yes. And my only comment is I wish we could have stayed with the current guidance. Thank you.
Thank you, Dr. Veda. Dr. Patel?
Hi, Kuldip Patel. I voted yes.
Dr. Gupta? Dr. Gupta, are you muted? Dr. Gupta, are you able to record your vote?
I voted yes. Hello?
Yes, we can hear you now.
Yes, I voted yes. Thank you.
Thank you. Ms. Fusco Walker?
Sandra Fusco Walker, I voted yes.
Thank you. With that, I believe we recorded everyone's vote. Dr. Stevenson, could you confirm?
Yes, I can confirm. Thank you.
Thank you. With this, we have ended the oxitriptan topic. We will take a 5-minute break before we reconvene at 5:20 Eastern Time for Neomycin Sulfate. Panel members, please remember that there should be no chatting or discussion of the meeting topics with other panel members during the break. Thank you. Welcome back, everyone. We will now have Dr. Takia Stevenson read the conflict of interest statement for this meeting's withdrawn or remove list.
The Food and Drug Administration is convening today's meeting of the Pharmacy Compounding Advisory Committee under the authority of the Federal Advisory Committee Act of 1972. With the exception of the National Association of Boards of Pharmacy, NABP, the United States Pharmacopoeia, USP, and the industry representatives, all members and temporary voting members of the committee are special government employees or regular federal employees from other agencies and are subject to federal conflict of interest laws and regulations. The following information on the status of this committee's compliance with federal ethics and conflict of interest laws covered by, but not limited to, those found at 18 U.S.C. Section 208 is being provided to participants in today's meeting and to the public. FDA has determined that members and temporary voting members of this committee are in compliance with federal ethics and conflict of interest laws. Under 18 U.S.C. Section 208, Congress has authorized FDA to grant waivers to special government employees and regular federal employees who have potential financial conflicts when it is determined that the agency's need for a special government employee's services outweighs his or her potential financial conflict of interest, or when the interest of a regular federal employee is not so substantial as to be deemed likely to affect the integrity of the services which the government may expect from the employee. Related to the discussions of today's meeting, members and temporary voting members of this committee have been screened for potential financial conflicts of interest of their own as well as those imputed to them, including those of their spouses or minor children, and for purposes of 18 U.S.C. Section 208, their employers. These interests may include investments, consulting, expert witness testimony, contracts, grants, quotas, teaching, speaking, writing, patents and royalties, and primary employment. During this session, the committee will discuss the revisions FDA is considering to the withdrawn or removed list. FDA is now considering whether to amend the rule to add one more entry to the list, Neomycin Sulfate. All parenteral drug products containing Neomycin Sulfate, except for ophthalmic or otic use, or when combined with polymyxin B sulfate for irrigation of the intact bladder. As previously explained in the Federal Register of July 2nd, 2014, 79 FR 37687 at 37689 through 37690, the list may specify that a drug may not be compounded in any form, or alternatively may expressly exclude a particular formulation, indication, dosage form, or route of administration from an entry on the list. Moreover, a drug may be listed only with regard to certain formulations, indications, routes of administration, or dosage forms because it has been found to be unsafe or not effective in those particular formulations, indications, routes of administration, or dosage forms. FDA plans to seek the committee's advice concerning the inclusion of this drug product on the list. This is a particular matters meeting during which specific matters related to neomycin sulfate will be discussed. Based on the agenda for this meeting and all financial interests reported by the committee members and temporary voting members, conflict of interest waiver has been issued in accordance with 18 U.S.C. Section 208 D3 to Dr. Kathleen Gura. Dr. Gura's waiver involves stock holdings of an affected entity. The aggregate value of the stock is between $50,000 and $100,000. The waiver allows the individual to participate fully in today's deliberations. FDA's reasons for issuing the waiver are described in the waiver document, which is posted on FDA's website at https:// www.fda.gov/committees/human-drug-advisory-committees. Copies of the waiver may also be obtained by submitting a written request to the agency's Freedom of Information Division at 5630 Fishers Lane, room 1035, Rockville, Maryland, 20857, or requests may be sent via fax to 301-827-9267. To ensure transparency, we encourage all standing committee members and temporary voting members to disclose any public statements that they have made concerning the topic at issue. We would like to note that Dr. Timothy Finsky is a representative member from the National Association of Boards of Pharmacy, NABP, and Dr. Jian Sun is a representative member from the United States Pharmacopoeia, USP. Section 102 of the Drug Quality and Security Act amended the Federal Food, Drug, and Cosmetics Act with respect to the Advisory Committee on Compounding to include representatives from the NABP and the USP. Their role is to provide the committee with the point of view of the NABP and the USP. Unlike the other members of the committee, representative members are not appointed to the committee to provide their own individual judgment on the particular matters at issue. Instead, they serve as the voice of the NABP and USP, entities with a financial or other stake in the particular matters before the advisory committee. With respect to FDA's invited industry representatives, we would like to disclose that Drs. Michael Booley and Gus Bassani are participating in this meeting as non-voting industry representatives acting on behalf of regulated industry. Their role at this meeting is to represent industry in general and not any particular company. Dr. Booley is employed by Cadman Pharmaceuticals, and Dr. Bassani is employed by Professional Compounding Centers of America, PCCA. We would like to remind members and temporary voting members that if the discussions involve any other topics that are not already on the agenda for which an FDA participant has a personal or imputed financial interest, the participants need to exclude themselves from such involvement, and their exclusion will be noted for the record. FDA encourages all other participants to advise the committee of any financial relationships that they may have with the topic at issue. Thank you very much, and I'm going to turn it back to the chairperson, Dr. Galore. Thank you.
Thank you, Dr. Stevenson. We will now proceed with the FDA presentation on withdrawn or removed list process from Gabrielle Cosell.
Good afternoon. I will provide a brief remarks on FDA's process for identifying drugs for the withdrawn or removed list. One of the conditions that must be satisfied for a drug product to qualify for the exemptions under Sections 503A or 503B of the Federal Food, Drug, and Cosmetic Act is that the compounder does not compound a drug product that appears on the list published by the Secretary of Drug Products that have been withdrawn or removed from the market because such products or their components have been found to be unsafe or not effective, and this is known as the withdrawn or removed list. A drug product that is included in the withdrawn or removed list is not eligible for the exemptions provided in Sections 503A or 503B. FDA periodically reviews available information on drugs withdrawn or removed from the market because such drugs or their components have been found to be unsafe or not effective, with the goal of identifying possible new entries for this list. The information we review may include Federal Register notices announcing withdrawal of approval of a new drug application or an abbreviated new drug application for safety or effectiveness reasons, or Federal Register notices announcing an agency determination that a drug product that was voluntarily withdrawn from sale was withdrawn for reasons of safety or effectiveness. We also review available information to determine whether any approvals of new drug applications would warrant modifications to existing entries on the list. In these cases, appropriate divisions within the Office of New Drugs evaluate each identified candidate or proposed modification using available information about the drug. And the responsible division will prepare a review of the information that documents its recommendations as to whether to include the drug on the withdrawn remove list, or remove the drug from the list, or modify an entry. FDA will update the withdrawn or removed list through notice and rulemaking as stated in a final rule published in October of 2016. We intend to propose regulations to revise the list when we identify drugs that we tentatively determine should be listed. And we also intend to propose regulations when we tentatively determine that changes to the status of drug products already on the list should result in a revision to their listing. And generally, we will finalize any additions or modifications to the list after consulting the advisory committee about the relevant drug and after providing an opportunity for public comments to be submitted on a proposed rule. As was mentioned today, FDA, we are discussing the following entries for potential inclusion on the list, and that is neomycin sulfate, all parenteral drug products containing neomycin sulfate, except for ophthalmic or otic use, or when combined with polymyxin sulfate for irrigation of the intact bladder. And that concludes my slides. Thank you.
Thank you. We will now proceed with the FDA presentation on neomycin from Dr. Jay Ho Hong.
Hi, good afternoon. My presentation is regarding the review of neomycin sulfate for the listing in the withdrawn or removed list. Next slide, please. First, I'll go over the review of withdrawn or removed list. Then I'll present the background of neomycin sulfate, including description, regulatory history, adverse reactions with parenteral use, and labeled indications. Then we'll address our assessments and recommendations. As a reminder, under Sections 503A and 503B of FD&C Act, FDA has established a list of drug products that were withdrawn or removed from the market because of these drugs have been found to be unsafe or not effective. Neomycin sulfate is an aminoglycoside antibacterial drug that was discovered in 1949. It is active against Pseudomonas, Klebsiella, Proteus, E. coli, and Enterobacter. It is poorly absorbed in the GI tract. The currently approved formulations of neomycin sulfate include oral tablets and solution, ophthalmic, otic, and dermatologic products, and a solution for bladder irrigation, which is in combination with polymyxin B. Following is the regulatory history of neomycin. Initially, neomycin was approved for intramuscular injection for serious systemic infections and urinary tract infections, intraperitoneal instillation for the treatment and prevention of peritonitis, intestinal instillation in abdominal surgery, and topical use. In 1972, labeling guidelines for neomycin sulfate sterile powder was amended, stating that the intramuscular neomycin sulfate may be indicated in the treatment of urinary tract infections, and due to its toxicity, limited the use to when other alternatives are not available. Also, the label included a box warning regarding nephrotoxicity, ototoxicity, and respiratory paralysis due to neuromuscular blockade. The Anti-Infective Advisory Committee meeting in 1977 concluded that there is no use of neomycin sulfate in sterile vials for parenteral use for the labeled indication, and that the risk-benefit for parenteral neomycin sulfate did not warrant its continued marketing. For the non-sterile neomycin sulfate bulk for prescription compounding, the committee concluded that a warning label should be placed. In 1979, FDA proposed to revoke provisions for certification of neomycin sulfate in sterile vials for parenteral use. Based on the findings of the Anti-Infective Advisory Committee, and to revoke provisions for certification of non-sterile neomycin sulfate for prescription compounding. Then in 1988, FDA revoked the provisions for certification of neomycin sulfate in sterile vials for parenteral use. FDA amended the provisions for certification of non-sterile neomycin sulfate for prescription compounding by changing the product name to neomycin sulfate for compounding oral products, and required labeling of appropriate uses and warning about the risks with inappropriate use. FDA withdrew approval of 4 applications for neomycin sulfate in sterile vials for injection in 1988. Also proposed to withdraw 6 applications for non-sterile neomycin sulfate products for prescription compounding, and subsequently withdrew 5 of these applications. The remaining last application was withdrawn in 2019 as the holder of this application waived its opportunity for hearing. The systemic exposure to neomycin sulfate by parenteral administration, including intravenous or intramuscular administration, instillation or irrigation of body cavities, structures, or spaces, or from use in wet dressings, may cause following adverse reactions: nephrotoxicity, irreversible ototoxicity, neuromuscular blockade, and respiratory paralysis. Following are the currently approved oral and irrigational neomycin sulfate products and their indications. First, neomycin oral tablet is indicated for adjunctive therapy in hepatic coma and as a part of regimen for suppression of the normal bacterial flora of the bowel. Neomycin oral solution is indicated as an adjunctive therapy in hepatic coma. Neomycin sulfate in combination with polymyxin B solution is indicated for urinary bladder irrigation of abacteriuric patients to help prevent bacteriuria and gram-negative septicemia associated with the use of indwelling catheters. The assessment of the approved formulations of neomycin sulfate, including formulations for oral administration, ophthalmic, otic, or dermatologic use, and in combination with polymyxin B sulfate for irrigation of the intact bladder, remains favorable. However, all other parenteral neomycin sulfate formulations may result in significant systemic exposure to neomycin and be associated with serious adverse reactions such as nephrotoxicity, ototoxicity, neuromuscular blockade leading to respiratory paralysis. Therefore, we recommend the following statement to be added to the withdrawn or removed list. All parenteral drug products containing neomycin sulfate, except when used for ophthalmic or otic use, or in combination with polymyxin B sulfate for irrigation of the intact bladder to be added to the withdrawn or removed list. This is the end of my presentation. Thank you.
Thank you. We will now take clarifying questions. For FDA presenters, please use the raise hand icon to indicate that you have a question, and remember to clear the icon after you have asked your question. When acknowledged, please remember to state your name for the record before you speak, and direct your question to a specific presenter if you can. If you wish for a specific slide to be displayed, please let us know the slide number if possible. Finally, it would be helpful to acknowledge the end of your question with a thank you, and end of your follow-up question with that is all for my questions. So we can move on to the next panel member. With that, do we have any questions? Seeing none, we will now begin the open public hearing session. Both the Food and Drug Administration and the public believe in a transparent process for information gathering and decision making. To ensure such transparency at the open public hearing session of the Advisory Committee meeting, FDA believes that it is important to understand the context of an individual's presentation. For this reason, FDA encourages you, the open public hearing speaker, at the beginning of your written or oral statement, to advise the committee of any financial relationship that you may have with the product, and if known, its direct competitors. For example, this financial information may include the payment by a bulk drug supplier or compounding pharmacy of your travel, lodging, or other expenses in connection with your attendance at the meeting. Likewise, FDA encourages you at the beginning of your statement to advise the committee if you do not have any such financial relationships. If you choose not to address this issue of financial relationships at the beginning of your statement, it will not preclude you from speaking. The FDA and this committee place great importance in the open public hearing process. The insights and comments provided can help the agency and this committee in their consideration of the issues before them. That said, in many instances and for many topics, there will be a variety of opinions. One of our goals today is for this open public hearing to be conducted in a fair and open way, where every participant is listened to carefully and treated with dignity, courtesy, and respect. Therefore, please speak only when recognized by the chair. Thank you for your cooperation. Speaker number 1, your audio is connected now. Will speaker number 1 begin and introduce yourself? Please state your name and any organization you are representing for the record.
Hello, can you hear me?
Yes, we can. Dr. Karam.
I'm Dr. Michael Karam, Director of Public Citizens Health Research Group. I have no financial conflict of interest. Slide 2. Regarding neomycin, Public Citizens strongly supports the FDA's proposal to add all parenteral drug products containing the aminoglycoside antibiotic neomycin sulfate, with the exceptions noted by the FDA, to the list of drug products that have been withdrawn or removed from the market because they have been found to be unsafe or not effective, and that therefore may not be compounded under the exemptions provided by Sections 503A or Section 503B of the Food, Drug, and Cosmetic Act, the withdrawn or removed list. Such actions should have been taken by the agency many years ago. Slide 3. In 1979, more than 4 decades ago, the FDA first proposed taking regulatory action to remove parenteral neomycin products from the market based on widespread evidence that the drug's risks outweigh its benefits. At that time, the fact that systemic exposure to neomycin caused serious nephrotoxicity, ototoxicity, and neuromuscular paralysis with respiratory arrest was well established, and no safe parenteral dosage regimen of the drug had been recognized. Moreover, neomycin was known to be more toxic than other FDA-approved parenteral aminoglycosides. Slide 4. In 1988, more than 3 decades ago, the FDA issued a final rule amending the antibiotic drug regulations to revoke the provisions for certification of neomycin in sterile vials for parenteral use, and another notice withdrawing the approval of 4 abbreviated antibiotic drug applications for neomycin for parenteral use because the risks involved in the parenteral use of neomycin were judged to outweigh any benefits that might be derived from its continued availability. Slide 5, please. In 1998, more than 2 decades ago, the FDA appropriately proposed including all parenteral drug products containing neomycin sulfate in its proposed rule to establish the initial withdrawn or removed list. However, in the final rule establishing that list that was issued in 1999, the FDA excluded parenteral neomycin products from the list because the agency had failed to take appropriate administrative actions to 1, address pending requests submitted in 1988 for hearings regarding the withdrawal of approval of applications for neomycin in sterile vials for injection, and the withdrawal of approval of the applications for neomycin for prescription compounding, and 2, respond to 4 petitions submitted in 1988 to stay these and other agency actions related to neomycin products. Slide 6. Disturbingly, the straightforward administrative actions for resolving these requests for hearings and petitions for stays of action related to neomycin products were not completed until February 2019, more than 30 years after these requests and petitions have been submitted to the agency. The FDA's decades-long delay in placing all parenteral drugs products containing neomycin, with the previously noted exceptions, on the withdrawn or removed list is unacceptable and borders on regulatory malpractice. The agency now must move swiftly to place parenteral drug products containing neomycin on the withdrawn or removed list, given their unacceptable risk-benefit profile. Slide 7. The following comments relate to FDA's process for updating the withdrawn or removed list. While the case of neomycin is an extreme example of unacceptable delays in adding dangerous drugs to the withdrawn or removed list, there are many examples over the past decade of drugs not being added to this list until at least several years after the FDA had determined that the drugs had been withdrawn or removed from the market because they had been found to be unsafe or not effective. Delaying such regulatory actions poses unacceptable and avoidable risk to patients and public health. Therefore, in April 2021, public citizens petitioned the FDA to take the following actions. 1, promptly amend through notice and comment rulemaking the withdrawn and removed list to include all drugs containing lorcaserin hydrochloride and all drugs containing the antibiotic bacitracin. And 2, promptly implement a policy stipulating that whenever the FDA issues a notice announcing a determination that a drug product was withdrawn from sale for reasons of safety or effectiveness, the agency simultaneously will issue an NPRM proposing to amend the withdrawn or removed list. Slide 8. Regarding lorcaserin, on March 4th of this year, the FDA announced in the Federal Register that the agency had determined that lorcaserin tablets marketed under the brand names Belviq and Belviq XR for weight loss were withdrawn from sale for reasons of safety or effectiveness, and that the agency would not accept or approve ANDAs for lorcaserin tablets. The FDA noted in its notice that results of a required post-market trial to evaluate the risk of cardiovascular problems suggested an imbalance in cancer in humans. Slide 9. The FDA further stated the following. Although chance effects cannot be ruled out, the imbalance in cancer persisted throughout multiple analysis approaches. The clinical findings corroborated by the evidence from animal models informed the agency's assessment that the risks outweigh any potential benefits for the current indications. These findings were considered clinically meaningful and could not be adequately addressed through labeling. Additional evidence would be necessary to investigate this signal. However, the agency has determined that it is unlikely that the necessary safety endpoints, i.e. cancer and reproductive safety, can be readily or ethically investigated in a clinical trial. Because preclinical and clinical studies would first need to be conducted to address these concerns, the agency has determined that this product would not be considered safe and effective if it were reintroduced into the market. Slide 10. Regarding parenteral drug products containing bacitracin, on March 12th of this year, the FDA announced in the Federal Register that the agency was withdrawing approval of 5 ANDAs for bacitracin for injection from multiple holders. The FDA noted the following in its notice. Bacitracin for injection is an antibiotic for intramuscular administration, the use of which is limited to the treatment of infants with pneumonia and empyema caused by staphylococci shown to be susceptible to the drug. Bacitracin for injection poses serious risks, including nephrotoxicity and anaphylactic reactions. Healthcare professionals generally no longer use bacitracin for injection to treat infants with pneumonia and empyema because other effective FDA-approved treatments are available and do not have these risks. In April 2019, an FDA advisory committee met and discussed the safety and effectiveness of bacitracin for injection. The advisory committee voted almost unanimously that the benefits of bacitracin for intramuscular injection do not outweigh its risks, including nephrotoxicity and anaphylactic reactions for the drug's only approved indication. The FDA notice reasonably can be read as a determination by the agency that bacitracin for injection was withdrawn from sale for reasons of safety or effectiveness. Slide 11. Moving forward, the FDA should not delay initiating the rulemaking process for amending the withdrawn or removed list once the agency has published a determination that a drug product was withdrawn from sale for reasons of safety or effectiveness. Instead, whenever the FDA announces such a determination, it simultaneously should issue an NPRM proposing to amend the list to include that drug product. Such simultaneous action could shorten the rulemaking process for amending the list by several months to years. Slide 12. Although Section 503A(c)(1) of the FDCA stipulates that the FDA must convene and consult with an advisory committee before implementing changes to the withdrawn or removed list, it allows the agency to issue such regulations before consultation with an advisory committee if it determines that doing so is necessary to protect public health. Such a determination certainly could reasonably be made in all cases in which the FDA has determined that a drug was withdrawn or removed from the market because it was unsafe, and likely could reasonably be made in most cases in which the FDA has determined that a drug was withdrawn or removed from the market because it was ineffective. Nevertheless, if the agency feels it must seek advice from its committee before issuing a final rule amending the withdrawn or removed list, the agency could schedule a meeting of the advisory committee for shortly after the NPRM proposing to amend the list is published. Slide 13 and my last slide. Such expeditious regulatory action would minimize the period during which patients could be potentially harmed by exposure to compounded formulations of drug products that were determined to have been withdrawn or removed from the market for reasons of safety or effectiveness. Delaying such regulatory action poses unacceptable and avoidable risks to patients and public health. Thank you for your attention.
Thank you, Dr. Karam. We will now take clarifying questions for the presenters. The open public hearing portion of this meeting has now concluded, and we will no longer take comments from the audience. Are there any clarifying questions of our presenters? Seeing none, we are now in the panel discussion phase and invite comments and discussion from our panel members. Seeing there are, I see Dr. Bassani. Do you have a question or a comment?
Yes, I do have a comment. This is Gus Bassani, industry non-voting member. Just speaking from the perspective of someone in the industry, I agree with FDA's recommendation. I'm not aware of neomycin being compounded into injectables or other parenteral dosage forms that would raise concerns. And FDA has clearly articulated that the addition to the withdrawn or removed list does not include ophthalmic, otic, topical, oral, and bladder installations with polymyxin B. So the wording is appropriate. Thank you.
Thank you, Dr. Bassani. Do we have any other comments or questions? Seeing as there are none, the committee will now turn its attention to address the task at hand, the careful consideration of the data before the committee, as well as the public comments. We will proceed with the questions to the committee and panel discussions. I would like to remind public observers that while this meeting is open for public observation, public attendees may not participate except at the specific request of the panel. Today's question is a voting question. Dr. Takia Stevenson will provide the instructions for the voting.
Question 5 is a voting question. Voting members will use the Adobe Connect platform to submit their votes for this meeting. After the chairperson has read the voting question into the record and all questions and discussion regarding the wording of the vote question are complete, the chairperson will announce the voting will begin. If you are a voting member, you will be moved to a breakout room. A new display will appear where you can submit your vote. There will be no discussion in the breakout room. You should select the radio button, that is the round circular button in the window that corresponds to your vote, yes, no, or abstain. You should not leave the no vote choice selected. Please note that you do not need to submit or send your vote. Again, you need only to select the radio button that corresponds to your vote. You will have the opportunity to change your vote until the vote is announced as closed. Once all voting members have selected their vote, I will announce the vote is closed. Next, the vote results will be displayed on the screen. I will read the vote results from the screen into the record. Next, the chairperson will go down the roster, and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. Are there any questions about the voting process before we begin?
Seeing as there are none, I will read the question into the record. FDA is proposing that neomycin sulfate, all parenteral drug products containing neomycin sulfate, except when used for ophthalmic or otic use or in combination with polymyxin B sulfate for irrigation of the intact bladder, be added to the withdrawn or removed list under sections 503A and 503B of the FD&C Act. Do you agree? Do panel members have any concerns with the wording of this question? Seeing none, if there are no questions or comments concerning the wording of the question, we will now begin the voting on question for neomycin sulfate.
We will now move voting members to the voting breakout room to vote only. There will be no discussion in the voting breakout room. Voting has closed and is now complete. Once the vote results display, I will read the vote result into the record. Voting has closed and is now complete. The vote results are displayed. I will read the vote totals into the record. The chairperson will go down the list and each voting member will state their name and their vote into the record. You can also state the reason why you voted as you did if you want to. There are 12 yeses, 0 noes, and 0 abstentions.
Thank you. We will now go down the list and have everyone who voted state their name and vote into the record. You may also provide justification for your vote if you wish to. We'll start with Dr. McElhaney.
Linda McElhaney, I vote yes.
Ms. Fusco-Walker.
Sandra Fusco-Walker, I vote yes.
Dr. Fensky.
Tim Fensky, I vote yes.
Padma Galore, I vote yes. Dr. Gura.
Kathleen Gura, I vote yes.
Dr. Bogner.
Robin Bogner, I vote yes.
Dr. Ravello.
Elizabeth Ravello, I vote yes.
Dr. Sun.
Amy Sun, I vote yes.
Dr. Patel.
Kuldeep Patel, I vote yes.
Dr. Veda.
Alan Veda, I vote yes.
Dr. Desai.
Jamal Desai, I vote yes, and would like to also point out I voted via the backup email mechanism.
Thank you, Dr. Desai. Dr. Gupta.
Dr. Gupta, and I voted yes.
Thank you. Dr. Stevenson, have we recorded all votes?
Yes.
Thank you. Before we adjourn, are there any last comments from the FDA? Seeing none, I would like to take this opportunity to thank the FDA for holding an excellent session and to all the presenters for their diligent work and presentation. It was extremely helpful to the committee as we made our decisions. I would also like to take this opportunity to thank all the nominator presentations as well as the public for their comments and opinions, which are of immense value in the decision-making process. And I'll end with thanking my fellow panel members. Thank you for your work today and for your patience as we work through a few technical issues and the delayed start. Again, thank you for everything from with regards to your efforts here. And I'll take a special moment to thank Takia Stevenson for her efforts in helping make this as smooth as possible for us as well as her team. We will now adjourn the meeting. Thank you.
The meeting is now over. All the participants have been disconnected.